It's very tough.
So this has never been easy since the onset of this work. So it is normal for it to be tough, but a lot of effort. It is well. I think today is our last meeting, right?
Please, yes.
Yes, so
Why yes, we're from the mafia.
no, it's nothing. No.
So by the
I'm
timetable
just
today,
sorry for myself. Looking
so
at what we have to read, and then I think next week we are beginning our quizzes or something.
Next
don't
week
think,
is
I think it's next 2 weeks, right? Because next week is for
Next 2 weeks.
practical. Yes.
practical.
Okay. Okay.
So you don't know your timetable. Shame on you.
Oh, say.
All right. So today we have 3 units to cover. 3 units, and as usual, it's our normal structures and the normal reason why some of the structures are preferred over others. So today I'll finish with the carbohydrates. I think I have some few slides on the carbohydrates. That is the lecture 6. Then after that, I will move straight to lipids or fat and oil. That will be the last slide that I will be talking about. So I think today we are enough. We have about 38 people here, so we don't need to waste a lot of time. Let me quickly move straight to whatever that I'm supposed to discuss. Well, last time we were looking at carbohydrates, and I think we've looked at the basic one, that is the monosaccharide. We spoke extensively on the issue of monosaccharides last week. So today I want us to move ahead and look at the other types or other forms. And the other forms, we have two main ones that we are going to look at. That is the saccharide and that of the oligosaccharides. Probably you also talk something small under what we call the polysaccharides. Remember when we started last week, I told you that when you have one glucose molecules, because it's one, it is mono. That is monosaccharide. I told you mono means one and the saccharide means sugar. So if you combine 2 monosaccharides together, you get what we call the disaccharide. And when they are around 3 to 9, we call them the oligosaccharides, and above 9, no matter how amount of glucose molecule that you can also combine with, we say that you have formed what we call the polysaccharides. And I think that one of the key things I told you that when these saccharides are being formed, there's a particular bond that does the work. It's a form of covalent bond. And the name of the bond is called the glycosidic bond. The glycosidic bond. So it is something that you need to know. And when you are putting 2 or more monosaccharides together to form a long chain of polysaccharides or oligosaccharides, or when they are only 2, that is the disaccharide, the bond that you need is glycosidic bond, and it's a form of covalent bond. And we know in our basic science that covalent bond mostly deal with sharing of electrons. It is the ionic bond or the electrostatic bond, whereby one particular compound or molecule just transfer its electron to one. Now with the covalent, there's always some form of sharing of electrons among them. So at the end of this lesson, I'm expecting you to describe what disaccharides are and how they are formed through what we call dehydration synthesis. Okay, so I'll spend a few times to talk about what we call dehydration synthesis. I know dehydration is just an English word, synthesis too is an English word, but we will talk about it. Dehydration synthesis. Anytime you get a word when you hear hydration, we are looking at something that's the water involved. Dehydration, that means that you are losing water. So sometimes when you in the afternoon when the sun is very scorchy and you are very thirsty, you say that, hey, I'm dehydrated. It means that you have lost a lot of, or you have lost a lot of water. So in chemical reactions, there are instances whereby certain chemical, certain compounds are combined, and at the end of the day, we lose water. And we will look at them. A typical example, we will look at how some of the monosaccharides are combined, and at the end of the day, they lose water. Now, we look at what is between alpha and beta linkage. So when these bonds are forming, there is what we call alpha linkage and beta linkage, and it's also had some important to some of the food that we consume. So the reason why you see some of the sugar compound very rigid, or we said that some of the carbohydrates are used for structural purposes. So anytime that you see that those that are used for structural purposes, it means that they contain what we call the beta, probably beta 1,4 linkage. So you look at alpha 1,4 linkage and beta 1,4 linkage, and we will just understand that. We will describe the function of different form of carbohydrate. Then also we look at some of the interaction of carbohydrate with other food constituents during food processing. So this will be our few objectives for the remaining slides of the carbohydrate. So this is the first thing that I want us to look at. I think you can have a look at the picture. Don't be scared. I'm just trying to use it to explain it. I think I will use this particular picture to do a lot of explanation even when I'm not on this particular slide. So just have a look at it and we will just talk about it. It's just sugar. You see, the first one is maltose and the second one the cellulose. So the first one is maltose, and you know that maltose is just a combination of two glucose molecules. So you can see that with the first picture where the maltose is, the first structure that you see there is glucose. The second structure is also called glucose.
Sir, please, we can't see you.
You can see my slide.
Yes. You're not sharing your screen.
I've shared the screen.
Is there?
My screen is shared. Please click from your end.
We can see it.
Yeah, I
Okay.
can see it.
Then check your network.
But thank you.
All right. So the first one that you see, maltose. Maltose is just a combination of glucose plus glucose. When you put two glucose together, it forms what we call maltose. That is, so maltose is a typical disaccharide. And I told you that when disaccharide is being formed, there is it goes through what we call the dehydration synthesis. Today, I think my voice is somehow down. It will come. I'm just tired. I stayed overnight. I decided to rest this morning, and I've realized that I have a class, so it will come. Let me move on. I can see that I'm not at my best, but it will come. You just don't worry. All right. So you can see that when this disaccharide is being formed, you can see there's a loss of hydrogen over here. The O that is in blue color. It is because it is being formed. We are losing that particular one. It is not only that one that we are losing. We will be losing some form of hydrogen. There will be some hydrogen here at the carbon one. And we say that the carbon one, the one that contains or that involves itself, let me say the carbon one of glucose is called what? If you remember, last week we did that. Carbon one in glucose is the one that mostly contain the what I made mention of specific. I'm trying to remember what I said last week. The carbonyl group, the carbonyl group is found on carbon 1. And I told you that when a carbon, the particular compound or the particular carbon that contains the carbonyl group is called what? I said it last week. I just want you to remember. But I will need it here. Yeah, it's who is talking. Efia, Becky, who is talking? Last week I made mention of one, Alima 2. I'm just looking at the name that is on my screen. Anita, Osei, Ula, SB. Yes, the carbonyl group of glucose is what?
Sir, please. The aldehydes.
No, please. The aldehyde is the functional group that it belongs to. So glucose belongs to functional group. So which particular compound contains that particular functional group? Which particular carbon? You know, the carbons are 6. You know, glucose molecule contains 6 carbons. And that is common to most of the sugars, with the exception of the pentose sugar that we've been talking about. So I just want to know that what is the name of that particular carbon? Hey, my brother. Yeah, that's that. Any idea?
No, I just joined this, so
I'm not asking today's question. It was last week's question that I'm asking, not today. Today you
last
not
week, last week I couldn't join. I traveled, so.
no problem. If I was better talking, you would have come for it. Yes.
Mhm.
Letitia. Any response?
Please say the question again.
The
Sir,
question
please, is it the aldoters or something?
very examine our income.
Is this the idle host?
Hey, you should be careful. You cannot mention words that does not exist.
Aldoses.
Aldose.
Still.
When we say aldose, what does it
Aldose.
Okay, don't worry. You remember them when you are writing your quizzes and all that. Some of them will come, and I can promise you that you people will be there and joke with it. Okay. So last week when we were talking, I told you that carbohydrate belongs to have 2 main functional groups. Then we spoke about the aldehyde functional group, and we said that we also have what we call the keto functional group. And I told you that mostly, as for glucose always belongs to aldehyde functional group. And I told you that the carbon that contains this particular functional group is what we call the anomeric carbon. That is the word I'm asking you people. Anomeric carbon. That is the first carbon. Aldehyde functional group, its anomeric carbon is number 1. But with the keto functional group, its anomeric carbon is number 2. Because that is where the functional group is basically found. Didn't I talk about this last week?
Yeah, you talked about it, but I don't remember you saying that. We just look at the placement of carbon group, which is the carbonyl. Yes, the aldehyde group. The carbonyl is placed on the first carbon, and the keto.
So
on the
the
second.
carbonyl group and the functional group, what is the difference? So because you didn't hear the word carbonyl, no, okay, then we will trick you people. Don't worry. All right, so that is basically what I'm looking for. And even the anomeric carbon is here. If you look at the second point, it said that it is the alpha anomeric or alpha anoma. That is C1. So the carbon 1, C1 is carbon 1. I'm trying to use it to, what I'm trying to tell you is that anytime it is going, the chemical reaction is going to happen. And there's these molecules, it will mostly happen either with carbon 1 and carbon 4. So most of the time carbon 1 and carbon 6. That is where we have the 1,4 linkage. We have alpha 1,4 linkage and beta 1,4 linkage. When you look at the top, when it is alpha 1,4, the what is it? The linkage or the bond is always pointed downwards. So you can see that with the first picture, the bond or the glycosidic bond. Remember, I told you that the bond that formed between two, this is equivalent, a type of covalent bond, and the specific one is the glycosidic bond. You know, glycosidic means the glucose, a bond that forms between glucose molecules. So just remember that. When it is pointed downwards, then it means that it is alpha 1,4 linkage. Alpha 1,4 linkage. When it is pointed in a zigzag way, can you see the second one is in the form of a zigzag? When you see that one, it means that it's a beta 1,4 linkage. And that is what I want you to understand. And there is a reason for that. When you consume food that contains a lot of 1,4 linkage, alpha 1,4 linkage, it is easy for your body to break it down to release energy. It's when you consume food that contains most of the time the beta linkage, our body finds it difficult to break it down. That is why this beta linkage is mostly found in food, the fibers and the cellulose and the rest. Those are the food that contain those particular linkage. And that is the reason why our body cannot break it down. The body cannot break it down because we don't have enzyme that can break beta. Most of beta linkage, we don't have enzymes in our system that can work on them. So these questions and this particular question and L and D glucose, they have a lot of things in common. Remember, we told you that D glucose is good for diabetic people because mostly the body cannot break it down, what and what and what. It's the same thing that we can also say that the alpha linkage, those disaccharides or the polysaccharides that contain beta linkage, the body cannot break it down. So those particular foods can also be good for people who are having diabetes. So the reason why those, the D glucose, it was just a mirror image of L glucose. It was the L glucose that we have just changed. We have done what we call the optical isomerism. We made mention of the optical isomerism of that. So we just change one particular, the arrangement. If it is on the right, we just change it to the left. That is how it has been done. And that's because of that reason, when the enzyme sees it, they see it to be a foreign material. This particular one, we cannot break it because we told you that enzymes are very specific in the kind of reaction that they go through or the kind of work that they do. So basically, that is, I just wanted to use this particular one to give you the explanation that, I spoke about two things, that anytime glucose is undergoing, bonding, when it's trying to form another compound with different molecules such as glucose, it uses its first anomeric carbon. The linkage is always about 1 and 4. That is the first carbon from first glucose and the fourth carbon for the second one. Then the linkage will occur. When the linkage occurs and the reaction, the linkage is pointed downwards, it means that it is alpha 1,4 linkage. When it is in the form of zigzag, it is beta 1,4. We also have alpha 1,6 and beta 1,6. All those ones are also there. Probably if you have time, then you think you can read on that. You can do so. There is a glycosidic bond. This is what I was talking about. Glycosidic bonds are bonds that join monosaccharides or longer sugar chain of other carbohydrates, forming what you call disaccharides, oligosaccharides, and polysaccharides. So anytime you want to form disaccharides, oligosaccharides, and that of the polysaccharide, your body, the kind of bond that is involved with glycosidic bond, very, very important. You must make sure you remember that. You say that these sugar molecules or the sugar chain of monosaccharide can form further glycosidic bond with alcohol and amide group to produce what we call acetal and glycosid and also the nucleoside. If you remember, I think I have made mention, we spoke about this acetal before. The one I was talking about the addition of alcohol to ketones and addition of alcohol to aldehyde sugars. We did it last week. I spoke about the acetal and hemiacetal. Then also, do you remember that when you add alcoholic or alcohol to ketone functional group, you get what you call the acetal and the hemiacetal, and when you add alcohol to the aldehyde functional group, you get what you call the acetal. I spoke about it last week. It's part of the slide that I gave it to you. So yeah, what the point is trying to say, we can still use monosaccharides or any glucose molecule can also do some further bonding with some compound that are not part of carbohydrate. An example that was given is the alcohol and the amide. Amide means amino acids. That is the amide group, and also to produce either the acetal or the glycoside and that of the nucleoside. Very, very important. We say that if the anomeric carbon of the sugar form the bond with oxygen atom in the hydroxy group in alcohol, the bond is made or it is named as O-glycosidic bond. So yeah, you need to pay attention to this particular point too. We say that anytime you are forming anytime the sugar molecule is forming a bond with any other compound aside the normal sugar molecule that's from any other compound such as alcohol, if the sugar uses 8 anomeric carbon, and remember that the anomeric carbon, we are talking about the carbon that contains the carbonyl group. So with the glucose, I've told you that 8 anomeric carbon or all the aldehyde, 8 anomeric carbon is found in carbon 1. It's anomeric carbon. And the ketone, an example of the aldehyde or those that form part of the aldehyde functional group, I made mention of glucose. And those that form part of the ketone functional group. I think the example I gave last week was fructose. So fructose is also a monosaccharide, and it is part of the ketone functional group. And the ketone functional group, 8 anomeric carbon is mostly at the second carbon, if I may remember. That is it. So we are saying that if the anomeric carbon of the sugar forms the bond with the oxygen atom in the hydroxy group in the alcohol, the bond is named O-glycosidic bond. O-glycosidic bond. That is the point that you need to remember. I will say that oppositely, that is the converse, the opposite side. If the anomeric carbon of the sugar form the bond with the nitrogen of the amide group, remember I told the point that we were making that either it's formed with the alcohol or the amide. When it reacts with the amide group, the bond that is formed is N-glycosidic bond. And we know that with the amide group, amide, the formula is the NH2 or something. When you see the formula, it contains nitrogen. That is why we are naming it here like that. So it is the N-glycosidic bond. And we say that, however, acetal, including those found in polysaccharide linkage, cannot easily become free aldehyde. It cannot become free aldehyde. That is acetal. You know acetal, when you are adding alcohol to that of the aldehyde functional group, that is what you form. And we are telling you that with that one, most of the time, it doesn't. You may not get free aldehyde, and when the aldehydes are not free, most of the time it cannot go through any form of bonding. They need to be that particular carbon that contains the aldehyde functional group should be free in order to undergo certain form of bonding. So that is another point that they also take it. So when we talk of disaccharides, this disaccharide is basically two glucose molecules or two monosaccharides that has been put together. Two monosaccharides. An example here is sucrose. Example here that we have here is sucrose. Sucrose is our sugar that we eat. We eat it in the house. Our table sugar. It's called sucrose. And we say that it is a common table sugar. It has glycosidic linkage, the anomeric hydrolysis of glucose and fructose. So when you see that the sugar that we are eating at home is not pure glucose, but rather it is sucrose, which is made up of glucose and fructose. Which is made up of glucose and that of fructose. So that is what we should know. I think let me use this particular one to talk about the dehydration synthesis. Before sucrose can form, sucrose go through a process called dehydration synthesis. That is that we synthesize glucose, or glucose is sucrose. Sorry, I'm using glucose. Sorry, it's supposed to be sucrose. Sucrose are supposed to form through the process called the dehydration synthesis. That is when sucrose is formed, water molecule is lost, or the reaction loses water molecules. And this is why I don't know whether you have paper and pen in front of you so that when I talk you can write. We all know that the basic formula for glucose and sucrose have their formula is the same. So we have C6, that is for glucose. We have C6, H12, and OH. That is the basic formula. Then fructose too have the same thing, C6, H12, and O6. So when you react this particular molecules together, you get C12. Please, I hope you are writing. Hello.
Please say you are moving fast.
I'm moving fast in terms of the writing or in terms of my talking.
The talking was.
So you're talking. Sir, please, can you take the formula again so that we can relate, please?
All right. What I'm saying is sucrose is a saccharide. And sucrose is our sugar that we use, and the one that when you are preparing your tea or whatever, you put it inside. That is what we call sucrose. And this sucrose is made up of two monomers. Let me use the terms. Two monomers. That is two monosaccharides. And the monosaccharides that are involved are the one that we call the glucose itself and fructose itself. Glucose and fructose. And I hope you know glucose belongs to which functional group? Glucose. It belongs to which functional group? Glucose. Which functional group? Is it aldehyde or
Aldehyde.
Keto functional. Idos.
Aldehyde, yes.
Aldehyde.
Glucose belongs. Mary. Mary, you were saying something. Oh, Mary, I can't hear you again.
Say,
I can't hear you.
say, please, I say keto group.
Glucose. Who support her? Who agree that it belongs to ketone functional group?
Say, I do.
You do. Who also disagree?
Please say I disagree. It's not.
I disagree. It's
Sorry, I
a
disagree.
high behind.
It is what aldehyde. Okay. I intentionally asked who agree with her so that I will know whether you all agree that it belongs to ketone functional group. No. The glucose belongs to an aldehyde functional group. That is what I said. And I told you that with the aldehyde functional group, 8 carbonyl group is found on the carbon 1. Then the fructose rather is the fructose that rather belongs to the ketone functional group. So you see here we have an aldehyde functional group coming to react with ketone functional group. And it's very interesting. And I told you that anytime they are reacting, mostly the ketone functional group will use the carbonyl group, which is carbon 2. Remember that. And also, the aldehyde functional group uses carbon 1, because that is where the numeric carbon is. That is the carbon that contains the functional group. And so remember that. So let's look at a picture. Let's look at a picture so that we can appreciate the theory that we said last week. So this is sucrose. The scientific name is there. We will mention it today. The actual name is there. So can you see that the bond is forming between carbon 1, which is in green in the glucose part, and carbon 2, which is red in the fructose part. So that is the point we are trying to make. Anytime they want to form this glycosidic bond, the bond will form between the the carbon that contains those carbonyl groups, and through that, water will be lost. So let's see this formula that I was trying to write. I said that glucose is having C6. I want you to write in front of you so that you appreciate what I'm saying. Glucose is having 6,6, H12, and O6. That is the formula for glucose. It's the same formula for fructose. Fructose is also having C6, H12, and O6. But when you react these two together, the final product, which is the sucrose, we are going to have C12, H22, and O11 plus H20, which is water. H2O. Let me put it, when I say 0, I'm thinking I mentioned numbers. So H2O. H2O is water. So that is how the reaction is. But you can see that when you check the carbon, the fructose is 6 and the glucose is also 6. So the final product we have 12. That is C12. When we check the hydrogen, it is 12 here. It is 12 for the glucose side. It is also 12 for the fructose side. But when we get to the sucrose, we have 22. So it means that 2 is mixing. Put it in mind. Then when you come to the oxygen, we have 6 here, 6 here. But when we came here, oxygen, we have O11. I mean, 1 is mixing. So the 2 hydrogen that is mixing, that is what has, the 2 hydrogen and 1 oxygen that are mixing in the formula, that is what has formed the water. So anytime disaccharide is formed, we lose its water. And that is the process that we call dehydration synthesis. That means you are synthesizing or you are producing disaccharide, but in the process, water is lost. Please, do you understand the point?
Please repeat it once again for me.
Okay.
You don't understand. What don't you understand? Because I'm just speaking chemistry. What we are saying is, anytime you want to form disaccharide, or when 2 sugar molecules want to come together to form a disaccharide, it go through what we call dehydration synthesis. It go through what we call dehydration synthesis. Put it in mind. Very, very important. When you hear dehydration means that you are losing water. In our normal daily life, we use this particular phrase that, oh, I'm dehydrated. What it means that your body has lost a lot of water. That is why you are dehydrated. Especially during the heat season, where the sun is very scorchy or the temperature is very high, you lose a lot of water through through your sweat. You need to have realized when the sun is too hot, you don't mostly urinate much, but you rather sweat more. But when the temperature is down, you rather lose water through urine as compared to through sweat. It's the mechanism that the body does. So once there is a lot of heat in the system, your body don't need to allow you to lose a lot of water through urine. Because when you are urinating, the water that comes out is much as compared to when you are sweating. So that is what you need to understand. So we lose it water, but in chemistry too, anytime you are combining two molecules of sugar together, you also lose water. And I gave you a formula that when you are combining, I gave you an example for sucrose, which is a sugar molecule. It's not only sucrose, that is sugar molecule, disaccharide. We also have maltose. Maltose is the combination of two glucose molecules. So you need to, I think we also have what we call lactose or something. That is the sugar that is found in milk, lactose. That is the sugar that is found in milk. So we can also have time and read on that particular one. I said that with the glucose, this is the formula, and I told you to write the formula so that when I'm explaining, you appreciate it better. So the formula for glucose is C6H12, C6H12, and O6. Then it is going to react with another molecule, which is fructose. So you bring the
Sir, please. Sir, please.
Vivian, I can't hear you. Vivian, you were saying something. What is it? Okay, I think she can also hear me. So let me continue. So I said with the glucose, we have C6H12 and O6. And the fructose also have the same formula, which is C6H12 and O6. And you know, in chemical reaction, when you want to combine these two, you bring plus in between them, plus. That is C6H12 O6 plus C6H12 O6. Have you all written that? Have you all written that? Vivian, you are back.
Yes, sir.
I wanted to say something. What is it? All right. So when you put this reaction together, what you are going to get is C12, H22, and O11. C12, H22, and O11, which is a formula, or which is a molecular formula for sucrose. Then after the C12, H22, and O11, you bring plus again. You bring plus again. Then you come and write the formula for water, which is H2O. If your hand was up, any problem?
Yes, sir. Please, with the glucose and the fructose, the C6H12 O6. I don't understand why you got H22, because when you add 12 plus 12, you get 24. So why 22? And then 6 plus 6, you get 12. Please, why 11?
You see, you have a legitimate question. But your question are supposed to be answered through the process of my explanation. So probably you understand it, you did the explanation better as compared to probably when I single it out to talk about it. Yes. At the final, you are supposed
to. Please, can I explain it to her?
Okay. My TA is
Let
here.
me
Becky,
try if I can.
Becky, talk for me. My TA is here. Becky, talk for me.
Yes, I was saying that the sucrose is when we the process of making the sucrose is the dehydration synthesis. And when we say dehydration, it's loss of water. So when you combine the glucose and then the fructose, there's a loss of water in that process. Therefore, the HO2 is the loss of water, or is the water that is lost in the formation of sucrose. So the H2 that is missing from the equation, and then the one O1 which is missing from the equation that forms the water that has been lost from the sucrose. That is why there's a reduction of H2 and then reduction of O1 in the formation of sucrose. That's the dehydration synthesis. So if I'm able to explain.
Can we all on our microphone and clap for her? Oh, your microphone is still off. Somebody, your microphone is still off. I think she has done that. Hey, Zulea, you didn't clap. I think she has done the best explanation for me. Yes.
Thank you, Madam Becky.
You understand it now?
Yes, sir. Yes.
Do you all understand it? I just want the whole class to just appreciate what she has said. That in the process of
Yes.
Yes, sir. Please understand.
Yes. So 2H will be lost, and also 1O will also be lost. That's the water. Thank you.
So you know, in our basic chemistry, where we were doing balancing of chemical equations, we said that the number of atoms on your right, or the number of molecules on your right hand, is supposed to be equal to the number of molecules on your left hand. So if they are not equal, it means that they are not balanced. Then you find a way to balance it. But when you look at our explanation that our sister gave, it's really balanced. So the two hydrogen that was supposed to be part of the 22, which will form the 24, is now the one that is part of the water, and the oxygen, the one that is lost. You know, we are supposed to have O12, but we have O11, and that is the one that is part of the water. That is the two. So, and we also have another synthesis that we call the hydration synthesis. Hydration means that when I want to, okay, let me use this one to just teach it again. Let me teach the hydration synthesis. I think at this point you will appreciate it better than when I use the same way. See, when you also have sucrose and you want to break it down to fructose and glucose, you are going to add water to that particular formula in order to do it. Do you agree with me? Now let's say,
yes, sir.
Yeah, when we were forming the sucrose, we have lost water. Water is no more part. So now when you want to get, when water has lost, so when you are writing the formula for sucrose, you are not going to add the water because that water has been lost. That's why we write plus water. It means that the sucrose plus water. It means that the water has been lost. That is dehydration synthesis. Now we have only the formula for sucrose, which is C6H22 and O11. Now I want to break this down to form fructose and glucose. How am I going to do it? Because when I look at these molecules in it, I'm going to short. As for the carbon, we won't have problem because 6 will be here, 6 will be here. But when I come to hydrogen, I'm going to have 11 here, 11 here. So there will be shortage of 2. Then when I come to oxygen, I will have 6 here and 5 on the other side. So it will also be short of 1. So if I want to get it right, I have to add water. I have to react water with this particular sucrose so that I'll be able to get my fructose back and I'll be able to get my sucrose back and my glucose back. And with that, we add water. And that is what we call hydration synthesis. That is, you are adding addition of water in order to create that particular compound that you are looking for. That is what we call the hydration. When you are drinking water, we say that you are hydrating your body. You are adding water to your body. That is hydration synthesis. Please, do you appreciate the difference between the two? Can somebody also speak about it, as our sister Becky spoke about the dehydration synthesis? Can someone also speak about the hydration synthesis for others to appreciate it? Hydration synthesis. Can someone try? hydration.
Please, sir, the dehydration synthesis, can you come again, please?
Zulea Mabu. Zulea Mustafa. Something that we have done
Hello,
and we are
sir.
moving on.
Sir, I lost my network, or I didn't get the time you were adding the oxygen. So I don't know. I don't know this chemistry. I don't know whether you said, I know we lost one oxygen. So will I do +0 alone or +0 1?
I don't know chemistry. We are all chemistry. We are all doing chemistry.
Oh.
Are you a home economics student proper? Or you are switching?
I'm switching. I'm a business student.
And you went and did your business and you did your account.
Yes, sir. Some of us are switching.
And today you are here. You are coming to the those of you who are here switching to our home economics field, go back to your business and go and teach your business.
Oh, sir, we beg.
This one, if it was FIFO, you would have easily understand.
Seriously, very serious.
You saw business courses, you decided to switch. You get to the secondary school, when the students are you tell them that you were a switch student. Okay. Auntie Becky, I think you have to come again so that Zulea will understand. I really love your explanation. Auntie Becky, are you there? Please don't come back again and tell me that you lost your network. I can't do anything. I told you that anytime you have to move from your village and come to town so that you get a network. Auntie Becky, come again with the dehydration synthesis so that your colleagues, some of them are still complaining.
With the dehydration. Please, are you saying the hydration or dehydration?
Dehydration. You speak about the dehydration again, and we will look at the hydration later on.
Okay. I said you were saying that with the dehydration, the sucrose. When we talk of sucrose, it means that there's a loss of water. That's the process of making the sucrose is the dehydration synthesis. And you said with the dehydration is loss of water. So, and when you combine the glucose, which is C6H12 and then O6 to fructose, they all have the same equation. So when you combine the two equations, we are supposed to get C12, H24, and then O12. But since there's a loss of water in the sucrose, the H is reduced by 2, and then the oxygen is reduced by 1. So therefore, the H2O forms the water that is lost from the sucrose. That is why we got the C12, the H22, and then oxygen is 11. Thank you.
Okay. Is Esme here? Today I've not heard of her. My personal assistant. Is Esme around?
Yes, sir. I just joined.
I hope you are doing well. I know you are not feeling well.
Yes, sir, by God's grace.
Okay. Is Ula here? Ula. Ula.
Please, I'm around.
Hey, another one go here.
You will feel.
All right. All right. So I think my sister has done it again. She has done explanation for me. I don't know whether Zulea, you are still having issue with that. The way she has done the explanation.
Yes, sir. Sir, thank you. I understand.
I have personal issue with Zulea. So when you ask me a question, I don't answer.
Sir, why?
Let's continue. So what we are saying is, once we have this dehydration synthesis, we may also have hydration synthesis. And I said that anytime there are certain reactions, they are reversible. Now we have reacted glucose and fructose together to form sucrose, which is disaccharide. You can reverse it back to getting your normal molecule. That is it. Glucose and that of the fructose molecules. And if you want to do it, because in the process of forming the sucrose, there was a loss of water. And when there's a loss of water, it means that we have lost 2 hydrogen molecules and 1 oxygen molecules. I've lost 2 hydrogen molecules and 1 hydrogen 2 hydrogen molecules and 1 oxygen molecule. So that is, so when we have lost it and we want to reverse the reaction to get our component, that is the fructose and the glucose, we need to add water to it so that we will be able to get our glucose and our fructose back. And the addition of the water to that particular reaction is what we call the hydration synthesis. That is, you are adding water in order to create those particular molecules that we have already lost. Is it better now? Can we differentiate between hydration, dehydration synthesis and hydration synthesis? Can we do that?
Yes, sir.
Aside Becky, can we do that? Can someone differentiate between the two for me? The difference between hydration synthesis and dehydration synthesis. Can we differentiate it before I move on? Yes. Yusuf.
Yeah, good morning, sir.
Good morning.
Yeah. With the dehydration synthesis, the water will be at the product site, but with the hydration synthesis, the water will be at the reactant.
Come again.
Site. So that is why.
Okay. I hope your colleagues understand the product side and the reactant side. You have used 2 big English words here. You have to break it down for them to understand. I get your point, but I want you to understand.
Okay, with products and reactants.
Yeah, you did talk.
Okay, like
you have a very poor network. I don't know where you find yourself. I can't hear you.
Like when you are going to do a so you say left side and hey. So can you hear me?
I can't hear you. I don't know where you find yourself. I can't hear you.
Oh. Hello, is it good?
Can you reposition yourself? Yeah, somehow better now. Talk and let me see.
So I was saying that with the products and then the reactants, like in a chemical reaction, we have the left side and then the right side. So the left side is the reactant side, and then the right side is the product side. So when we talk of the dehydration synthesis, the water will be at the right side. That is the product side. And then with the hydration synthesis, the water will be at the left side, and that is the reactants side. I think it's clear.
Okay. It's clear, but I think I understand your concept where you are coming from. I really appreciate that.
Okay.
When you are asked, probably you find yourself in an exam hall and you are asked that differentiate between dehydration synthesis and hydration synthesis. And probably you put it in that perspective. Well, maybe a teacher will always, the person or the examiner will surely understand what you are saying. But probably you may not fetch you the full marks that you need to get. Because when you are trying to differentiate between these things.
With the dehydration synthesis, we all know that the formula for the glucose and the fructose is 6 C6H12 and 06 plus.
Gloria, let me finish with what I was saying. I will come back to you. Don't worry. So what I was expecting you probably to, because most of the time when you are asked, you are not probably going to draw the structures for them. You are going to probably write it. And you know how when we ask you to differentiate something, when the question doesn't ask you to use table, probably you need to use certain conjunctions such as while. Probably this and this involve this, while this one and this involve this. So you can talk about it in that perspective. But whatever you said here is right. At the reactant side, the reactant, when we want to produce sucrose, the reactants are the glucose and that of the fructose. They are the reactants. Then the product becomes the sucrose. The sucrose and that of water. That will become the product. So I understand you. Gloria, come in.
So, say, when we talk about the dehydration, basically it means that we are doing away with water or removing water from the product. So we know that our glucose is C6, H12, and 06. And the fructose is also 66, H12, and 06. Now we want to get a product which is the sucrose, which becomes
let's use the 06. For 0 number.
06. So when it reacts together, the combination of the glucose and the fructose, which comes to form the sucrose, which is the C12, H22, and 06, plus water, which is the H20. Now, when it comes to the hydration, we want to reverse it back so that we'll still get our water in the process or in the formula. So the loss of the water, which is the two hydrogen molecules, which react with the sucrose, will give us our glucose and the fructose back, meaning that we still have water in the product.
Yes. So is that an explanation or you are differentiating among the two?
You say we should differentiate.
Oh, okay. I like it. I'm impressed. Any other person to talk about it?
So please, the next question.
If you complain with the issue of
It's not
network, I'll
helping.
fight you. I'll ask you that anytime you have class, move from your village. Even if it is a tree that you have to climb, you climb it. And you can react like a Tigo advert. After that, you just get down and go. Sophia, are you here? Today, I want the class to be more interactive, so I will not rush through. I want everybody to talk. Sophia. I can see Sophia. I know a lot of people are not part of my class, but once you have come here, they're entitled to talk. Sophia. Sophia, your network is good. Don't tell me you can't hear me. Then Vida. The Vida, I have Vida. Otiwan. Vida. Anything to say about that? So basically your colleagues have given the differences. So when you talk about the dehydration synthesis, in my own view or in my context, if I want to differentiate it, I will say that dehydration synthesis is a chemical reaction where two molecules are joined together with the removal of water. A chemical reaction where two molecules are joined together with the removal of water or water molecules. Whilst hydration synthesis is a chemical reaction where two molecules are split into, when a molecule is split into two by addition of water, you can also say that it is a chemical reaction where water is used to split a molecules into two. When water is used to split molecules into two. So that is basically a few understanding that we need to understand. So when you consume sugar in your diet, Let me bring it here so that you can appreciate it. When you consume sugar in your diet and get to your stomach, there is an enzyme called sucrase. Sucrase. You know, we have sucrose. The enzyme that breaks sucrose down is called sucrase. The only difference is the O in front of the R is A here. There's an enzyme called sucrase that will break this particular molecules down into fructose and sugar and glucose. And our body can absorb glucose and fructose because they are monosaccharides. So our body will just absorb them into our bloodstream so that the body will use it to generate energy for itself. So when your body do not have that particular enzyme called sucrase, your body cannot work on that. So that is how it is. It's the same thing like when you consume maltose. When you consume maltose, your body contains an enzyme that we call the maltase, and the maltase will break the maltose down into glucose and that of glucose. You know, maltose is two glucose molecules, so the body can break those down. Please, dehydration synthesis. You also call it condensation reaction. Those of you that you have a little knowledge in chemistry, it's also condensation. Condensation is that you are bringing two together. When you are putting things together, you are condensing it. Condensation reaction. And also the hydration synthesis is also called the hydrolysis or hydrolysis reaction. That is the hydration reaction. You also call it the hydrolysis. Hydrolysis, that is, you are using water, water. So these are some of the things that you can just use to appreciate this particular concept. I think I've spent a lot of time here. Let me move on. So you can see that the glycosidic bond is forming between the carbon 1 and the carbon 2. And the reaction is pointed downwards. So when it's pointed downwards, it means that it is alpha 1, 2. It is alpha. One is pointing downward, it is alpha. So one is alpha, you look at the carbon that are involved in the reaction, then you call alpha 1, 2 leakage or alpha 1, 2 glycosidic bond. So this is the full name for sucrose. So sucrose, the name for sucrose is alpha D glycoparanosyl. It's rather glucoparanosyl. 1, 2. So that is the name. That is the full name for sucrose. This one is not something we are going to ask you. Just I want you to appreciate that when you see it somewhere, you should be able to appreciate how it is. Another disaccharide is here. We have milk sugar. I think I have spoken about this. We have milk sugar, which is lactose, and lactose is composed of galactose and glucose. So when you put one glucose molecule and one galactose, you produce what we call lactose. And this one, we said that the linkage is who can name this linkage? I think the name is already even there. So you can see that the linkage is beta 1,4 because it's in the form of zigzag. Beta 1,4. It is forming between the glucose is using the numeric carbon, which is 1, and the galactose is using the 4. So beta 1,4 linkage, and that is what is happening here. Let's continue. Now let's talk about oligosaccharides. That is from 3 when your molecules are more than 2, 3 up to 10 or 9. Most of the books also say 9. You are going to get what you call the oligosaccharide. So that one is more, the chain is longer than the disaccharide. So we say that they are covalently bond. That is, the bond that also exists between them is covalent bond. And we say that they can also occur whereby a glucose molecule can be reacting with a protein. That is what we call the glycoprotein or a lipid, which we call also that one as a glycolipid. So these glycoproteins, the glycolipids are something that you made mention when we are talking about when we get to the fat side. So one of the examples is the raffinose. We said there is an oligosaccharide found in peas and beans. So there are some amount of sugar that is found in beans and peas, and the name is the raffinol. And we say that they are mostly undigested, so they reach the intestinal flora in the large intestine. Which releases hydrogen, carbon dioxide, and methane. Methane is a form of gas. Methane is a form of gas that is mostly released. So we know that in our basic digestion, we are told that most of the cellulose, some of them pass through the digestive system without being digested. So when they get to the large intestine, there are some bacteria over there that we call them the intestinal flora. So they will act on it and release hydrogen, carbon dioxide, and sometimes methane. And most of these things that you see here, they are gases. So sometimes that is the reason why when you consume, there are some people when they consume a lot of those fiber foods, they experience some kind of gas within their stomach. And these are some of the reasons. So put it in mind that when we talk of the oligosaccharide, we are looking at when you have a long chain from 3 to 10 monosaccharide that has been covalently joined together. And the bond that also exists is mostly the glycosidic bond. That is what is being used to link them together. So we don't want to waste my time here. So let's talk about polysaccharides. So polysaccharides is when you have more than 10. So from 11 and above, it means that you have moved to polysaccharide. We said that polymers consist of chain of hundreds or thousands. So can be hundreds of thousands of monosaccharides. Or sometimes you can have monosaccharides and disaccharides. So you can have glucose, galactose, then you come and see sucrose, then you have lactose. Or lactose and sucrose are disaccharides. So you can have monosaccharides and disaccharides that have been covalently linked together. So you need to understand. Because at the end of the polysaccharides with an anomeric carbon, one is not involved in glycosidic bond. It's called reducing end. So you see, when you have a glycosidic bond, when you have this polysaccharides, the last molecule will not be bonded to any sugar or any monomer. And that particular one, because it is not bonded to any monomer, it is having the anomeric carbon open, and that is what we call the reducing end. So most of the time we said sugar is glucose is a reducing sugar, and that is where the concept also we got the concept from. And we said that polysaccharides are not reducing sugar. No, polysaccharides is not a reducing sugar. Since the anomeric carbons are connected through glycosidic linkage, anomeric polysaccharide, most of them, the anomeric carbon which contain the carbonyl group is always going through bonding. That is through the glycosidic linkage. Let me bring the picture here. So you can see that the anomeric carbon is always bonding, and once the anomeric carbon has been bonded to another one, it cannot go through what we call the reducing end. It cannot have the reducing end because the anomeric carbon is already in the process. Is already bonded to another monomer that is in front of it, so it cannot be said it is a reducing sugar. It cannot be done unless probably the last one. So that is what we can say about it. So let's look at some of them. Some of the nomenclature of monosaccharide. When we say that we can have polysaccharides that are homopolysaccharides, who can differentiate between them? This one, I want someone to just talk about it. Homopolysaccharides and heteropolysaccharides. I want someone to speak to it because it's an English word. I don't want to talk about it because it's an English word. Hetero and poly. Hetero and poly. Homo and hetero. That is the difference I want to draw here. Homopolysaccharide and heteropolysaccharide. Yes, who will talk about it for me? Or as you mentioned names. Yes, Mavis. I can see your microphone.
Except the homo
Saccharides is talking about same sugars. One sugar, and the hetero is talking about the different type of sugars bonded together.
Good. You are good. You are a chemistry student. Next week you will teach chemistry. All right. So what we are saying is that anytime you hear the word homo means that the same type of something. So when we say we have homo polysaccharide, it means that we can have only glucose molecules. And Alima too, you wanted to say something. Your hand is up. Yeah, Alima Ibrahim. Okay, let me continue. So you can have only glucose molecules, about 1,000 of them that have been linked together by a glycosidic bond. And that particular one, we call it homopolysaccharide. Homopolysaccharide. Then when we come to the hetero means that different type of monosaccharides or different type of monosaccharide that has been linked together. That is what we call hetero. So you can have glucose, you can have sucrose, you can have lactose, you can have fructose that has been linked together. We call that one as what? As hetero. Hetero means different type. Hetero. So that's why you have homosexual. Homosexual means that a man and a man, a woman and a woman. So those of us that we go in for a man and woman and woman and a man, we are heterosexual. So we have homosexual and heterosexual. So that is how this particular concept can also be understood. I hope you all get it. Are you all okay with that particular concept? I think we are. Silence means consent. So with the polysaccharide, we have those. We have some polysaccharide. The example of polysaccharide, we have starch and glycogen. And we say that starch and glycogen are storage form of carbohydrates. Starch and glycogen, they are the storage form of carbohydrates. So I prefer someone to talk about it. Which of them is found in plants and which of them is found in animals? How does animals store carbohydrates and how does starch store carbohydrates? Yes, today is our last day, so everybody will have to talk. Yes, Matilda, are you there? Matilda, how does animal store carbohydrates? And how does plant also source carbohydrates? Animals are the human being. We are part of the animals. Yes, we are animals. Yeah, we are just the highest form of animals. Yeah, Matilda, you wanted to say something, Matilda?
No, you called me and I responded, please.
You want to say something on the starch and how can we differentiate between starch and glycogen? Starch and glycogen. Yes, peace. I have seen peace. Yes. Yeah.
Is for the plants and then glycogen is for animals.
The starch is for the plant and
yes.
Glycogen is for the animal. Yeah, that is perfect. That is good. You know, plant stores their sugar in the form of starch, and animals, including human beings, we store ours in the form of glycogen. So that is what you need to talk about. That is why when someone is eating a lot of carbohydrate food, you say that, oh, you are consuming a lot of starch. Yes. So when you look at photosynthesis, when photosynthesis occurs, when we were doing photosynthesis way back in school, we said that photosynthesis is the process whereby plants manufacture their own food with the use of probably oxygen and the energy from the sunlight, which we call the chlorophyll. So the chloroplast will trap the chlorophyll and combine it to manufacture their own food. And when you manufacture this food, store it in the form of starch. So let's use cassava for example. Cassava is a green plant. You plant your cassava, the cassava will germinate, it will grow. Then the plant, the flowers will attract this sunlight and combine it with oxygen and stuff and produce energy. when this energy or the food is produced, it is stored inside the tuber, the one that is inside the soil, so that you go and uproot it. When you uproot it and you go and cook, you get your food. So plants store their energy in the form of starch, and we human beings and other animals, we store our food in we store our carbohydrates in the form of glycogen. So if you have remember, when we are testing for photosynthesis in the lab, we test for starch. Once we are able to identify that a starch has been formed, it means that photosynthesis has occurred. When photosynthesis occurs, the only evidence we will know that that particular plant should have some amount of starch. So you said, I think if I could remember, you use an iodine solution and you add it to the leaf. And when the color that's supposed to come is blue-black precipitation. When you see blue-black precipitation, it means that photosynthesis has occurred, which is very, very important. So we the animals, we store our hours in the form of glycogen. So this morning you have consumed a lot of carbohydrate food, a lot of sugars. Then your body has used the part that it wants. There are excess. When there are excess, the body tries to store them. And in our body, there are two main places that the body uses to store starch. The first one is the liver. The liver stores some amount of glycogen. Then after your liver, your muscles. The muscles. So people who go to gym and stuff, they eat a lot. The reason is they have a lot of muscles. And these muscles need energy in order to lift their metals and do the heavy exercises and stuff. So this is the two ways that you can differentiate between glycogen and that of starch. Starch is a storage form of carbohydrate in plant, whilst glycogen is a storage form of carbohydrate in animals. So when you are fasting as individuals, it will get to a point that your body will start breaking down these glycogens to give you energy. Sometimes it break it down to raise your sugar level, and sometimes you break it down to just release an energy. So most of the time, the glycogen that is stored inside your liver, is basically used to monitor your glucose, your blood glucose level, so that it will not go down. You know, when your blood glucose go down, the term that we call it, the hypoglycemia. That means when it go too much down, you can die. And we also have the hyperglycemia. When you see someone suffering from hyperglycemia, it means that the person having diabetes, too much sugar level in your blood. So we need to just understand these two basic concepts so that we will be able to know. That is how carbohydrates are stored. They are stored in the form of sugar, glycogen in animals, and starch inside the plant. So let's know that. They also have what we call the chitin and the cellulose, and we say that these ones are structural molecules. So when you look at, I think last week I made mention of this, that we human beings, we possess what we call endoskeleton. Our skeleton is inside our body, and it is covered with our flesh. It is covered with flesh. But there are other animals that they have what you call the exoskeleton. That is, their skeleton is found outside their body. An example is cockroach. Cockroach is having exoskeleton, and those structures, skeletons are very hard. They are hard in nature because they are there to give some form of protection and structure. Those exoskeletons are made up of polysaccharides. They are made up of polysaccharides. You can also pick plants. The outer surface of every plant is very hard, and that is what we call the cellulose. These are all part of polysaccharides that form it, and you call those ones like structural polysaccharides. Structure. So chitin, you can talk of most of the animals that are having exoskeleton. Crustaceans, those that they are found in sea. All those ones are made up of polysaccharides. They are polysaccharides. So the first one that I spoke about, we say that starch. It is a plant storage polysaccharide. It is polymer consisting of D-glucose unit. It is usually insoluble in water because of the high molecular weight but they can form a thick colloidal suspension with water. So you see, starch is a D-glucose unit. And what does it mean? When we say D-glucose, what does it mean? What are the properties of D-glucose? Yes, D-glucose. Yes, D-glucose. Hey, move me. D-glucose.
So that's saccharide.
Then hey, last week, you remember L-glucose and D-glucose? Do you remember L-glucose and D-glucose? Huh?
So the D-glucose, yes, we remember.
What did you say about it?
So you said that the D-glucose, they are found in humans and animals, and then they all are found in fruits and like plants. And then
I spoke to you extensively. I want more information about what you said about D-glucose and L-glucose.
So the D-glucose is the main source of energy for all living organisms.
Good. So basically, that is one point. D-glucose, you know, we said that the D-glucose are the main source of energy for almost all organisms. And we know that we human beings, we eat a lot of starch. Our rice that we eat, our cassava, our corn dough, the banku that we eat, all the glucose that is found in them, most of them are D-glucose. That is why they are. But when it comes to the cellulose and the fiber and co, those ones contain what we call the L-glucose. So that is a few things that we can just look at it. And we are saying that they are insoluble in water. It means that they cannot be dissolved in water. That is one. So you remember when you are doing your last week, I used an example that when you are doing starch, when we were young, we use cassava, then we would just grind it, squeeze the water out, and use it to prepare starch. You know, when you put those starch in water, it will become very difficult for it to dissolve. But rather, it will form suspension. It will just form a very thick substance and just be suspended on top of the water. All right, so that is a few things that you can. All right, that is a few things that we can just look at it. Then we said that we have two main forms when it comes to starch. We have two main forms of starch. We have the one that we call the amylose and the amylopectin. Amylose and amylopectin. Today, since I've wasted a lot of time here, I need to move on. Please, are you following? I hope I'm not running faster. Are you okay with the pace?
Yes, sir.
So, are you okay with the pace?
Yes, sir.
All right. So we have the amylose and we have the amylopectin. Okay. So we say that most starch, that is 10 to 30% are amylose and 70 to 90% are amylopectin. So the amylose, the typical amylose is when you have a long chain and on branch. When the chain is just a linear, we said it is amylose. But when you have a branches within the chain, we call it amylopectin. So these ones they are all there to also give a specific food for some of the conditions that we experience when probably we are growing up. So when you talk about amylose, this is an example of amylose. Look at the picture up there. That is a long chain of monosaccharide, long chain. It doesn't have any branches. It is an amylose. So when you consume this particular food, amylose, they can easily be digested by your body. They can easily be digested because there is no branches. So the enzymes will just find a way to break the bonds, the glycosidic bond that exists between them, and to release individual glucose or fructose molecules for your body to use it to perform its function. So we said amylose consists of long unbranched chain of thousands to 2,000 molecules, consisting of alpha 1,4 linkage. And I think I have spoken about this. Alpha 1,4 means that the bond are forming between a numeric carbon 1 and that of carbon 4 of the other molecule continue like that. So the chain is being formed. And we say that 0 to 20% of starch in plants is in this form. Yes, that is why our body solely rely on this as a form of energy, because we have the enzyme, abundant enzyme to break this particular ones down. And we say that amylose form helices, or it form a coil when they are trapped in a molecule of iodine, forming characteristics of deep blue, purple color. So the first thing I was talking about something, this is where it is. It is because of the amylose that we are able to test whether photosynthesis has occurred or not. So anytime amylose find itself when iodine is around, it form a blue purple color. Blue purple color. And once a blue purple color is formed, it means that there's a presence of starch. And once there's a presence of starch, it means that photosynthesis has occurred. So this was what we've been doing in the lab. So you see, it's related to food. So once you are cooking, you have to also know how to test for some of these things. All right. Then also we are saying that the alpha-1,4 glycosidic linkage are more easily digested by enzyme. Yes, they are easy to be digested by enzyme. We have a lot of enzyme in our system that can break down this alpha-1,4 linkage. Let's look at the opposite side. That is amylopectin. So the only difference is that you can draw here is with amylopectin, they are highly branched. We have a lot of branches. These are few ones. When you see the picture of it, we have a long amylopectin which is having a lot of branches. And we say that consists of a long chain of glucose up to 105 molecules connected by alpha-1,4 glycosidic linkage, alpha-1,6 branches. Every 24 to 30 molecules unit, there is a branch. So that is what we are saying, that when you are counting them, when you get to every 24, so if 1 to 24, there will be one branch. From 24 to maybe 48, there will be another branch. From 48, then you move on, another additional 24, there will be another branch. That is what the point is trying to talk about. And we say that 80 to 90% of starch in plants form from this. And we say the branches produce a compact structure and provide multiple chain ends, which enzymatic cleavage occurs. So if the enzyme want to break down this particular one, they have to break it down from where the branches are. Because when you break the branches, you are going to get a linear one, or amylose. When I break this, look at where my casa is. When I have an enzyme that is able to break this particular one, I'm going to have two amylose, because this one is a long chain, or this is a linear chain. The second one too is a linear chain. So know that the amylose are mostly, if someone come to my end that the person want to reduce weight, I will recommend the person to eat a lot of amylopectin food rather than amylose, because amylose can easily be broken down in our body. But with amylopectin, because the linkage, the amylopectin have two linkages. That is the alpha-1,4 and that of alpha-1,6. But because of the 1,6 here, the breakaging is not easy as compared to the alpha-1,4. So though the body can break it down, but it will not be very quick as compared to the amylose, which contain only alpha-1,4 glycosidic linkage. Then the glycogen that we spoke about, I said that glycogen is a storage form of starch or sugar in animals. And I think I've spoken extensively on that. And it also contains alpha-1,4 linkage and alpha-1,6 branches. They are highly branched. They also have a lot of branches. So you need to just understand that. Then glycogen is even more branched. However, branches occur in every 8 to 12 glucose units. Remember, with amylopectin, we said that it occurs in 28 to 30. Every 28 to 30 glucose unit, there is a branch. But with the glycogen, it occurs at 8 to 12 glucose units. And we said that glycogen is abundant in the liver and the muscle. So I spoke about this. I said that the main two parts of the body that stores glycogen is your liver and your muscle. The liver one is used for blood glucose level, or to monitor the blood glucose level, and the muscles ones are used for energy production. Then we said that on hydrolysis, it forms glucose, which maintain normal blood sugar level and provide energy. So can someone talk about hydrolysis here? When we say in hydrolysis, what does it mean? What does it mean? In hydrolysis, it will form glucose. What does it mean? Any idea? In hydrolysis, glycogen will form glucose. What does it mean? I want someone to think about this and talk because I've spoken about it. What does it mean? Hey, Mongaza. Or I should put it in a different perspective. Do you understand what hydrolysis means?
Sir,
Do you understand what
please.
hydrolysis means?
Sir, is it the breaking down of the compound, the chemical compounds?
Do you people remember that? You people just remember that I made mention that what is it? Dehydration synthesis is also known as condensation reaction, and hydration synthesis is also known as hydrolysis.
So it means when water is involved in the reaction. Go.
So, yes, you are having a point, but what I'm expecting to know is glycogen is just a lot of different molecules of glucose that has been put together and stored. And you know, when a lot of glucose molecules react to each other, we lose water along the way. So in hydrolysis, if you want to break glycogen back to get individual glucose molecule, you need to get water. You need to add water to the reaction in order to produce the glucose molecule that we want. That is why we say that on hydrolysis, it forms glucose. On hydrolysis. That means that when it goes through hydrolysis reaction, and hydrolysis reaction is the same thing like the hydration synthesis. Hydration synthesis. I spoke about this. I was expecting a lot of you to just, your mind should just get to this particular point for me. But fine, let's continue. So we said that on hydrolysis, it forms glucose because the body cannot use glycogen energy in the form of glycogen. They have to break it down to glucose. Then the body will absorb the glucose. Then it will go through the pathway that we spoke about, the glycolytic pathway, the pentose phosphate pathway, and the Krebs cycle, and the rest. It go through those and it produce energy in the form of ATP. So know that. So we said that when they are on hydrolysis, when it is breaking down into glucose, it helps to maintain your normal sugar level. And remember, I told you that most of the time, the glycogen that is stored in the liver is the one that maintains the sugar level, and the one that is found in the muscles produces energy used to provide it. And there's a reason why the one that is stored in the muscles cannot be converted to free glucose to give energy. And there's a reason. If you have time, you can go and read on them. There are particular enzymes that your muscles are lacking. That is why you cannot perform that particular function. So let me go on. Then we also have the dextrans. So the dextrans, we said that change in the main linkage between glucose from alpha-1,4 to alpha-1,6. Alpha-1,4 to alpha-1,6 resulted in a newly formed polysaccharide dendran branch. So we are saying that we can also change the linkage that exists between, most of the glucose molecules is having alpha-1,4 glycosidic linkage. So when you change it to alpha-1,6, alpha-1,6 mostly have the branches. So when you change it, then you form what we call the dextran. And we said that they are found in bacteria and also component of dental plaque. So if you see somebody who is having a bad teeth, that's the dental plaque. There's some discolor on their teeth, or the teeth has been discolored. Those brownish nature or the brownish color you see on their teeth, that plaque contains some amount of dextrans. It means that they are all carbohydrate units that we have just changed the linkage that exists between them from the alpha-1,4 to alpha-1,6, and that results in that particular color that you possibly see it right there. So this is the structure of the dextran that I'm just talking about. They are found in bacteria and also the dental plaque. So let me talk about cellulose. So cellulose, we're saying cellulose fiber. So let me say cellular is a polymer consisting of a long unbranched chain of D-glucose connected by beta-1,4 linkage or beta-1,4 glycosidic linkage. Remember, I've been telling you that the alpha-1,4 are those that the body can easily break it down, but the beta-1,4 sometimes it's difficult for the body to break this one down. And that's why we are saying that because of the beta-1,4 glycosidic linkage, making it difficult for mama to break it down. So when you consume this one, you feel your tummy this one, but mostly your body cannot. You feel your tummy but mostly your body cannot use energy. Becky, can you mute yourself? You are giving me feedback. Mute yourself at my end. You are giving me feedback at my end. All right. So let me continue from. So we have the cellulose. They are made up of beta-1,4 glycosidic linkage, and that is the reason why we cannot. Our body find it difficult to break it down. You said that it consists about 300 to 3,000 glucose units in one molecule. So when you pick one cellulose, it contains a lot. So when you pick let's say you are eating orange and you just finish squeezing the juice off that particular roughages that you see. They are part of cellulose. When you pick one alone contain about 300 to 3,000 glucose units. And we said that it's a structural polysaccharide. I think I've spoken about structural polysaccharides already. The one that is some of the animals who are having the exoskeletons, that is what it is made up of. Most of the exoskeletons are made up of this particular cellulose. And we said that it's most abundant in nature. So it is very common, and its principal strength and support to tree and plants. Most of the time, they give support to trees. So when you see a tree, all the outer covering of the tree are cellulose, and they give support and strengthen the tree so that it can withstand with any of the harsh environment that it finds itself. And also, because of the beta linkage, cellulose has a different overall shape from amylose, forming extended straight chain and hydrogen bonds on each other, resulting in very rigid structure. So what they are saying is because of the beta linkage that is between them, it makes it become more rigid. Rigid means that it is very hard in nature. It is very hard in nature. If you have seen most of these crustacean skeleton that I'm talking about, the exoskeleton, they are somehow hard. And the hard nature of it is because it has a way of bonding with other hydrogen, going through having another hydrogen bonds to each other. That make it more rigid as compared to the amylose, whereby it's only involved in glycosidic bond. There is no more hydrogen bond that is in between them. All right. So we're also saying that the most animals lack the enzyme needed to digest cellulose, although it does provide the needed roughages to stimulate the contraction of the internal system and help food to pass through the digestive system. So this point is just telling you the importance of consuming fiber. And we say that one of the important is it helps in digestion, the movement of digestion. Fiber, one of the important is it helps in digestion, it helps to prevent constipation, and also sometimes it also prevent colon cancer. A lot of fat deposits are stored in your intestine. It can lead to colon cancer, but when you consume a lot of fiber, because they are heavy and when they are moving through your intestine, you can clear some of these fat deposits in your intestine. Then we said that other structural polysaccharides, the chitin. I think I spoke about chitin. It's the exoskeleton of crustacean, insect, and spider, and the cell wall of most of these animals. We are saying that they are made up of chitin. We also have the arganids, that is carbon binding polymers in an algae. So when you get to algae is also having polysaccharides are their structure. And we have the agalose and agalopeptin. That's galactose polymers. When you have a lot of galactopolymers that are also forming the agalose and agalopeptin. And we have glycoamino glycans. That is repeated disaccharide with amino acids. When you have a lot of amino acids, that has been bonded with sugar with a negative charge, it forms what we call glycoaminoglycans, and these are all structural polysaccharide that we can talk about. Should I talk about this? I think I was speaking about this last week. Last week I spoke about Maillard reaction, right? Did I teach Maillard reaction last week?
So yeah, you spoke about it.
Yes, I spoke about
it. You didn't teach, but you just spoke about it.
I gave you you said I didn't teach. I gave you the definition and the importance. Is that not true?
Oh yes.
So I'm trying to teach. I remember what I did last week.
You did.
You are laughing. You think I don't remember? I didn't show me. So I spoke about Maillard reaction, and I said that Maillard reaction is a reaction that exists between. Let me keep quiet and you people will talk about it. So what did you say? Let's talk about Maillard reaction. Let's some few minutes so that we can start with we have 2 hours more so that we can start with our facts.
Sir, please, you said that the Maillard reaction, when there is a reaction between reducing sugar and amino acids due to heat, which produces a desirable characteristics in food, such as the golden brown color found in baked foods, and then aroma of roasted granules too. That was what you said.
All right. I think basically this is what you need to know at this level when we come to Maillard reactions. Probably last week I spoke about only the desirable characteristics. Probably there may be an instance whereby Maillard reaction can also produce undesirable characteristics. And I think it is not core component of your course outline, but if you care to know, you can just read about negative effects of Maillard reaction. So Maillard reaction, basically we are looking at when there is someone, I'm just talking about it before someone just sent me a message that some people are new here and they have to they want to also understand. They're like, I don't want to be wicked. They were supposed to be here last week, so once they are not here, it is not my duty. So Maillard reaction, we say that most of the time when we roast our food or we bake our food, we experience some characteristics that is very desirable. When you say desirable, that is the one that we want, very favorable to us. That is what we call the Maillard reaction. So when that particular aroma that you see, the colors that you see in roasted food and baked food is as a result of the reaction between amino acids, specifically reducing sugars, and that of amino acids and reducing sugars in the presence of heat. So those reactions, this result that you see, that's a result of that. So we have a lot of amino acids reacting with glucose molecules. And at the end of the day, to produce those desirable characteristics. So when you are marking, when I come to your school and I'm marking your baked product, these are some of the things we look out for. At least a cake should be golden brown, it shouldn't be dark in nature, golden brown. Then I know we all know how when you are baking, some of the aroma that are supposed to come. And this aroma that you see, it doesn't come by surprise or by magic or miracle. No, it's as a result of the components. So when you have a lot of glucose and amino acids molecule within the food, it is going to produce a lot of these desired characteristics that we are looking for. And we are saying that it's observed in grilling, browning of meat, and formation of crust in baked products. Sometimes when you go to the brewery, that's when they are brewing their beer or roasted of cocoa and coffee and beans. All these ones are as a result of that. You can also get it from when we are roasting garnet. And we say that in addition to the darkened color, many complex flavors are also developed. There are a lot of flavors that are developed when it comes to Maillard reaction. And we say that there are hundreds of compounds produced during this reaction, not many of which are well characterized. So a lot of them may not have specific characteristics that you can use to discuss them. We are done with carbohydrates. Any question? Any question? Any question on your microphone? Let me hear from you before I move on to fats. Any question on carbo? Any question on carbo? Monosaccharide, disaccharide? We are done with all those things. Any question? Oh, let okay. And then let me quickly continue. I'm a good student and I'm happy for that. Very, very good ones. My people, are you there?
Yes, sir, we are here.
Can you see my screen?
Yes, sir, we are here.
Yes, sir.
Have you seen my screen?
Yes, sir. We have seen chains
Yes,
of.
sir. Yes, sir.
Yes, sir.
You are seeing some Latin structure on your screen, right?
Yes.
Don't worry. You are supposed to understand the structure and you will understand it. Don't worry. Okay, so we were looking at the macronutrients. I think the first one we spoke about protein. I just finished with the carbohydrates. Now let's quickly talk about. Let's talk about the lipids or the fat and oil. You are in class and you are having your own conversation. Momo, you.
Sorry, sir.
All right, so let's quickly look at fat and oil. So we have crucified fat and oil for a long time, and fat and oil is not good. Me, I don't want to be fat. If I take in fat, I will do this. I will increase in weight. This and this and this. All the macronutrients are very, very important. You cannot ignore any of them. All of them are so important. That is what I would say. Fat has a lot of role that it plays in our system. Without fat, most of you cannot give birth. If you don't have an amount or substantial level of fat in your system, you can't give birth. Or this fat or this lipid. And yes. That is why a lot of, if you are too slim, when you are underweight, giving birth become a problem because most of your sex hormones, those hormones that involve in your menstrual cycle that help you to ovulate, those that help you to the process of fertilization and stuff are produced by fat, specifically cholesterol. I don't want to take it. It contains a lot of cholesterol. Yes, without the cholesterol, to most of us, not even only the female, even with the issue of male, without some level of cholesterol, male, our hormones is called, I think, the testosterone. And you ladies, you have a lot of estrogen in your system. Estrogens are mostly dominant one that regulate your sexual characteristics. When these hormones are not there, you cannot produce enough. A man, you cannot produce enough. What is it? Spermatozoa, because those what is it? This cholesterol, which is made up of fat, are those that are involved in what is it? What we call the sex hormones in human beings. So we shouldn't crucify it much. Probably the only problem is too much of them become a problem in our system. When you have a lot of fat in your system, it becomes a problem because fat is a rich source of energy. It is a rich source. It contains a lot of energy than any other macronutrient that we've looked at. So that is a general thing that I will say about fats before I will delve into it. So we should know how to classify them, differentiate between saturated and unsaturated fatty acids. Then we talk about the essential and the non-essential fatty acids, and how we can name them based on their orientation and number of double bonds that they form, and where the double bonds are also placed in the fat. It's also something that probably is to be of interest to us. When we talk of fat, we are saying that they are just a group of substances that are generally soluble in organic solvents, but insoluble in water. So fat cannot be dissolved in water, but it can be dissolved in organic solvents such as turpentine, such as kerosene. Those ones are inorganic solvents. So those ones can dissolve fat. So most of your paint that we use to paint our doors and our homes contains a high level of fat. That is why if you want to mix it, you use turpentine. You don't use water. The water can only dissolve the emulsion paints. Emulsion paints contain a lot of water. But when you look at the oil paint and stuff, because of the oil nature of it, we use turpentine and those things. So these things that we normally do at home, there are reasons behind them, probably because we don't know them. We say that they are mostly made up of carbon, hydrogen, and oxygen, and some may have an addition of phosphorus, nitrogen, and sulfur. We have heard of phospholipids and the rest. They are lipids that have been combined with phosphorus, then it has its own function that it plays in our system. The classification, we can have fatty acids. We can classify fats as fatty acids, the glycerol, the phospholipids, the waxes, and the sterol. So the sterol are those that form part that produces the sterols are. I think a typical example of sterol is the cholesterol, the one that I've been talking about. So sterol is very, very important in our system. They regulate all our sexual characteristics that we've been talking about. I would say that the main classes of lipids found in food found in food and in the body are the phospholipids, the sterol, and the triglyceride. So these are the three main ones. The three main ones. We have the phospholipids, we have the sterol, then we have the triglyceride. So most of the fats that our body store the fat in particular from the triglyceride to the triglyceride. So I will show you a structure of the triglyceride in the course of this particular lecture. We are saying that with the lipids, about 2% of dietary lipids are phospholipids. So about only 2% of all the lipids that we consume from our food are phospholipids. And we say that they are found in food of both plant and animal origin. And in the body, the body can also make some of the phospholipids. So what you need to understand that there are instances where your body can produce. There are a lot of things that your body can do it on its own. That is why sometimes those that the body can produce it or synthesize it by its own, you have to reduce the intake of it because it may become an excess. And also some research also shows that the body produces it when it needs or when the body wants to use it to perform specific functions, then it go through some form of synthesis to produce those ones. Is it that unlike lipids, phospholipids are soluble in both water and fat. Yes, when you check the structure of phospholipids, it has a hydrophilic part and hydrophobic part. I think this thing, we remember this thing when we were doing our proteins. We spoke about hydrophilic and hydrophobic. So anytime they find themselves in water and fat environment or oil environment, they form what you call a bilayer, lipid bilayer, so that it will be able to interact with both water and that of the fat. And we said that only small percentage of our dietary lipids are sterile. That is most of them. The most familiar type of animal sterile are what we call the cholesterol. So most of the time, cholesterol is mostly, we normally get them from the animal origin. So that the body also make cholesterol, which is important component of cell membrane, precursor in the synthesis of sex hormones, adrenal hormones, vitamin D, and the bile acid. So pay attention to this. You can be asked to give about 2 important of cholesterol in your system. So cholesterol help your body to synthesize sex hormones, and we have the adrenal hormones, the cortisol. Then you can talk of vitamin D and the bile acid. You know, the bile. We have what you call I don't know how to put it. When you kill an animal, there is a bile. In Tree, you call it bonino. I don't know, those of you that you don't understand, you forgive me. There's some greenish substances that is in a sac that is attached to the liver. We call it bile, and the bile, its function is to help in, to digest fat. So in digestion, we call it the emulsification of fat, that is breaking down of fat. That particular bile contains an amount of acid, and that acid is being produced by what do you call the cholesterol. So you need to understand that, produced by cholesterol. And we say that the largest category of lipids is fat and oil, which is also referred to as triglyceride. So you can see that when we talk of lipids, we are not looking at only fat and oil. There are other components. What is it? The phospholipid I made mention of, they are not fat and oil, but they are lipids. The waxes, the sterol, which is made up of, which is the common sterol that we know is cholesterol. All these ones are lipids, but they are not fat and oil. And also fat and oil is a component of lipids. And the term that we use to call fat and oil in food is triglyceride. Triglyceride, that is, I think that is the first picture that I show you. I'll get there, I'll talk about it. Please, are you following?
Yes.
Are you with me?
Yes, sir.
Yes,
All right.
sir.
Am I moving faster?
Yes, so we
Right.
want to write something since, so we are moving too fast.
But in himself, we are better taking one.
You are writing. What do you want to write? So that I'll read it again, then you write.
The salient
So I
point.
think afterwards you go first one after the other, or that is all.
That is all.
Hey, sir, let me say your introduction.
Hey, sir, calm down.
There.
I'm not on top. I'm already down. So.
No, you're on top.
So explain that. So you are giving us the importance of the cholesterol. So I was able to write only one.
What did you write?
I wrote it. You said it.
Sex hormones.
I know that is what you write, because that is what is interested to you.
Oh, so I say.
All right. So what I was saying is, we have a lot of, when we talk of lipids, it is a group of different compounds. It's a general group that has been given to that. And lipids consists of fat and oil, which we call it the triglyceride. That is fat and oil. It also consists of sterol. And I don't want to say sterol and cholesterol, because cholesterol is an aspect of sterol. The sterol, we have different types of sterol, but cholesterol is an example of sterol. We also have what we call the waxes. Then we have fatty acids, and we also have glycerol. So these are all molecules or compounds that we name them, or we term them lipids. I think phospholipids is also one. And remember when I was talking about phospholipids, I said that the phospholipids is able to work or act best in watery environment, and also that is a hydrophobic environment and hydrophilic environment, because it has this particular end. It contains those ends. When you look at the structure of it, it has hydrophobic end and hydrophobic end. So when they find themselves in an environment whereby it contains fat and oil, they are able to work on that. Even the phospholipids, they are part of our cell membrane. You know, we've been saying that our cells are semi-permeable. When we are doing plant cells and animal cells, we said that animal cells contain what we call cell wall, and these cell walls are semi-permeable. When you hear the word semi-permeable, it means that it allows certain substances in and out of the cell. And this semi-permeable nature of the cell wall is as a result of the phospholipid that is found. It is found in it. So that is what we can say about. Please, someone is at my door. Let me attend to the person before she continues to disturb me.
Stella.
Efia,
Who campus ana?
after na meko.
All right, I'm back. Okay. So I was talking about this phospholipid that it is. I think this thing that I'm saying, it is better you write them, because when you ask, for example, a function of phospholipids, it's something that you can search a lot about it and write them. But the typical one that we can talk about is those that it form part of our cell membrane, which regulate the amount of substances that enters the cell or that come out of the cell. So these are some of the few things that we can just talk about. Then I was talking about the last point, and I told you that the body can, with cholesterol, we get some only few percentage of cholesterol is gotten from our diet, and that one we mostly get them from our animal origin, and the body can also make its own. That is why we caution you not to consume a lot of cholesterol, because the needed one can be produced by the body itself. So why should you consume a lot of them? And we say that cholesterol is also very important when it comes to cell membrane and a precursor. When you hear something is a precursor, it means that how do I even put it? Precursor means that is what the body uses. It's a precursor. It's an avenue for the body to use to synthesize the sex hormones. The sex hormones. So aside the sex hormone, they give us adrenal hormones. Example is the cortisol. Adrenal hormones, that adrenaline, when you are in fear. But now when those of you find yourself in your room, when you hear that your room is burning, you can manage your way to pass through the window and get out of the house. But at the normal circumstances, when your house is not burning, you cannot run away like that. Sometimes you'll be surprised that, hey, I never knew I can run like that. Sometimes when you are in fear, that is where you discover your talent. So you can just find yourself in one and discover your talent and stop. Probably you can stop learning. Okay. We are also saying that the largest category of lipids is fat and oil. So when I ask all these ones that I'm talking about, the major part of lipids are fat and oil. They constitute the most. They are many in abundance. The fat and oil, and we refer to them as the triglycerides in food. Triglycerides. So that is what I've been saying so far. So those who said they are writing, I hope you've been able to write it now. Have you been able to write it now?
Yes,
Those
sir.
who were writing. Okay. So we are saying that fat and oil are made up of individual molecules called fatty acids, which differ in the chain length. I think I have a picture that I will show you to you. So let me just go on. We say that the short chain fatty acid have 2 to 4 carbon atoms. Medium chain have about 6 to 12, and that of the long one have about 14 to 18 carbon atoms, while a few have more than 20 carbon atoms. So that is what you need to know, that we have the long chain, the short chain, the long, and the medium, and a few ones that can have more than 20 carbon. So anytime you hear this triglyceride, this is the picture of triglyceride. Let me show you to you. This is a picture of it. Triglyceride is made up of 3 molecules. It consists of 3 molecules, 4 molecules in general. It consists of 3 molecules of fatty acids and 1 glycerol. That is what we call the backbone, the glycerol backbone, which has been esterified together. So you can see that this is the glycerol. That is where my Keza is, the glycerol. This is this particular one. The C that is bonded to a 2H and O. And when you come here, the metal 2 is having the same. C bonded to O here and H here, and C bonded to 2H.
Please, we can't see.
Check your network. Well, I know others can see. Check your network. Please, I hope you can see.
Yes, sir.
Yes, sir.
Yes, sir.
So you can see where my Keza is, right?
Yes, please.
All right. So let me continue. So where my Keza is what we call the glycerol, and it's always the backbone of the triglyceride. Remove my picture. If you remove my picture, you will see the structure. Have you seen it now? Those of you who are using phone, you know if you don't take time, you can flip this particular screen off and come up with the others. So you need to know how to use the phone that you are holding, because probably a lot of them can see, and me myself, I'm even teaching from my screen. I've also joined the link with other phones so that I know whatever I'm doing, you people can see. Can we continue?
Yes, sir.
All right. So that is what we call the glycerol. That is the backbone. It is the backbone of every triglyceride that you can think of. Then we also have individual fatty acids. So we have one fatty acid top here, another fatty acid, and the third fatty acid that has been joined together. So this is just the structure of triglyceride. So you can see that some part contain double bond and some part contain single bond. And so you need to just understand that. So you see the first one here, there's a double bond, there's a double bond, and there's a double bond. There's a reason for that. As you move on, we will get to understand. So basically, this is how fatty acids are. Okay, so we have the polar part of it, and we have the non-polar. So the R chain, when they are reacting with non-polar region, and this is the OH. Anytime you see OH, it means that water. That is, they are hydrophilic in nature. So these are just some of the structures that I just want to use it for you to appreciate it. And also fat and oil or fatty acids can also be grouped into what we call saturated and unsaturated. And this point is very, very, very important when it comes to we home economics students, those of us into nutrition. Saturated fatty acids and unsaturated fatty acids. So the key thing that you need to understand here is when you hear the word saturated, anytime you have saturated fatty acids, when there is a room temperature, that particular fatty acid is supposed to be solid in nature. So any oil that becomes solidified at room temperature, it means that that particular oil is saturated. Then those that are always liquid at room temperature, those ones are always unsaturated. They don't tend to become solid, that they are unsaturated. Now there's one difference that you can draw from saturated fatty acids and unsaturated fatty acids. I think you can jot it down. It will help you one way or the other. And the saturated one becomes solid at room temperature, and the unsaturated one are always liquid at room temperature. Then the saturated fatty acids don't contain any form of double bond. their bonds are always single. The bond that exists between the carbon, that, it is carbon, that has been linked to hydrogens. What is it? Carbon that has been linked to hydrogens. But with a saturated one, the bond that exists between them are always a single bond. There's no double bond that form between them. So that is what you need to just understand. Then when you come to the unsaturated one, they have double bond, and some of them may even have the triple bond in it. They have the double bond. And that is what you need to just understand. But under the unsaturated fatty acids, we have the monosaturated and the polyunsaturated. I think once we've talked about mono, we should just understand. Mono means that it contains only one double bond, and the poly means that it contains more than one double bond. That is two or more. Please, do you understand? Do you get the point I'm trying to make over here? Saturated and unsaturated.
Yes, please.
All right. And what I want you to know is, most of the oil that we are supposed to consume are those that are unsaturated. So we go to the market, they say this is coconut oil, this is sunflower oil. They are all unsaturated fatty acids. Because the saturated ones are not good for our health. So when you kill an animal, those of you that you just finished celebrating your salad without giving me meat, when you prepare your soup, and you leave it overnight, and the next day when you pick the soup, you can see that the animal fat has been solidified. It is an example of saturated fatty acids that has been solidified. It means that it contains only single bond that has been solidified. And those particular oil are not good for our consumption. That is why we advise people to limit the intake of those particular fats, those particular fats. But the ones that are monounsaturated or polyunsaturated, they don't solidify easy. So they are good for our consumption. They don't solidify easy, so they are good for our consumption. Palm kernel oil, palm oil, and the rest, they are those that we want them. And even the unsaturated carbon, fatty acid, they also have the arrangement of their chemical formula, or the molecular formula is cis. It's a cis isomer. So if you remember when we were doing the isomer. Here we have cis isomer. When it is cis, it means that all the carbons all the hydrogens are arranged in a straight chain, So we have two main types. We have cis and we have trans. I will talk about them. The cis isomers are the best, and they are most of the natural occurring ones are cis isomers. Cis are the best. When it becomes trans, then it becomes a problem. So if you have heard of trans fat and the rest before, this is where the ideas, or we get these ideas from. So we need to be just mindful that the cis ones are the best for our system, and the trans ones are not good. So that is it. So we said that we have one double bond that is under the monounsaturated, one double bond. They are widespread in living world where they occur mostly as cis isomer. Then the poly one, they have two or more double bonds, especially conjugated fats. So that is how we can classify fatty acids as saturated and unsaturated, to put it in mind. Then fatty acids can also be classified as essential and non-essential. And I know this one from our knowledge back at proteins. We know what essential things and non-essential ones are. So the non-essential ones are those that it can be synthesized in the body that we made mention of. And it includes saturated and omega-9 fatty acids. Omega-9 fatty acids. And in the course of the day, I will tell you the reason why we call it 9, omega-9. It is telling you that probably the double bond is found at the 9th carbon. That is why we name it as omega-9 fatty acids. and it does not matter if our diet do not contain them. So when we don't even consume omega-9, our body can produce it. And we have the essential ones. So we are going to do some pronunciation exercise here. So everybody should on the microphone. We are going to do pronunciation exercise. Yeah,
Hello,
because, yes.
sir. Please, you said the omega-9, is it essential or non-essential?
It's non-essential. Your body can produce it.
Okay, thank you, sir.
All right.
Sir,
Yes.
sir, please, you said that omega-9, the reason why it is called omega-9 fatty is because the carbon is on. I did not get that, please.
Oh, I said that, you see, we call it, we have omega-5, omega-6, omega-3, and omega-9. The omega-9, I told you that this unsaturated ones, they contain double bond. And omega-9, the double bond is found at the 9th carbon. The 9th carbon, when you are arranging it, the 9th carbon contains the omega-9. That is, they contain the double bond. That is why we call it omega-9. We also have omega-3 and omega-6. All right. So with the essential fatty acids, we said that the body cannot produce them. So it is important for us to include them in our diet. And we said that it includes fatty acids with carbon-carbon double bond before the 9th carbon. So before the 9th carbon, when you are counting from the meter end, so you know that. And we say that the body cannot manufacture omega-6 fatty acids or omega-3 fatty acids. The omega-6 fatty acid, we call it the linoleic acid. So let's all say linoleic acid.
Linoleic acid.
I want to. So the day that you
Linoleic.
I can hear linoleic.
Linoleic.
That's like
Is it have money to turn you people into KG students? I've been able to tell people's wives into. So we have linoleic acid. That is the omega-6. Then we also have the omega-3. That one is called linoleic acid. See it.
Linoleic acid.
Those of you that you have off your mic. And your microphone is off. And
linoleic acid. Linoleic acid.
Okay, off your microphone. I think you people you like to be in KG environment than at the university. So let's continue. All right. So we said that with the linoleic acid, the body cannot manufacture it. That is the omega-6, and the linoleic acid too. That is the omega-3. The body cannot manufacture them. So we need to consume them in our diet. So that is very, very important. So if you have seen most of these, this your sunflower oil that you see in the market, they will be writing it contains omega-6 and omega-3. Sometimes they do advert on TV and they will tell you omega-3 and omega-6. They are very, very important. And one of the food stuff that, if I may remember, is salmon. When you consume salmon a lot, this particular fatty acids are common to get. So my Adventist people who don't consume it, probably may find different type of food that you may also get them from. Very, very important. So we said that with the sources, so they are even here. with the sources, that is omega 3. That is the omega 3, the food that soya bean oil and the walnut and omega 6. We can get them from the seed, the nuts, and that of common vegetables. Common vegetable and fish and shellfish. You can also get those things from them. Now we said that phospholipids are abundant in egg yolk, liver, soya bean, and peanut. So I made mention of phospholipids, if you remember, and I told you that they are responsible for the movement of material in and out of a cell. So you can get phospholipids from your egg yolk, you can get them from your liver, and also you can get some from your soya beans and some peanuts. Peanut is also called the granules. Then another fat or another type of lipids that we spoke about earlier was the sterols, and I saw that cholesterol occurs in food of animal origin. So most of the time it's not easy to get cholesterol from plant origin. Most of the time we have it from the animal origin. So I told you people that there are two main nomenclature of unsaturated fatty acids. And they are trans and cis. They are trans and cis. At the natural order, we were supposed to, the cis ones are those that our body wants. The cis one. And we say that cis or trans, then we have given you, what is it? They have given you a particular formula here, a triangle and a box. So the triangle here is trying to tell you where they exist. I think it will be, we will explain it when it gets there. We say that the double bond is located on the S carbon. Carbon bond, counting from the carbonyl terminal. The cis or the trans notation indicate whether the molecule is arranged in a linear or in a branch form. Remember I told you that the cis is always arranged in a straight line, that is in a linear form. But with the trans one, I don't know why I didn't take a picture of these things to show you to you. It's unfortunate. I will try and see if I can get a picture of it to show you. We are saying with a cis type, let me see, I'm trying to get a picture to show you. With a cis, the cis is having the cis is having the arrangement of the cis. I think I'm trying to get a picture here to show it to you. Blah, 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 blah. I'm coming there, just give me a minute. I think I have one here. Let me just copy it so that you can appreciate it better than just talking about it hypothetically. Can you see what I've done? Can you see it?
No, sir.
Can you see my screen?
No. I'm going to screen it up. No.
Yeah, you can see your screen, but it's not.
Okay, I'm coming.
It's the old one that is on it.
Okay, I'm coming. I hope you can see the new one now.
Yes, sir.
Yes, sir.
You can see the structure that I
No, I can't see.
Try and see. All right. I think I've placed some structure over there. Everybody should be able to see. I can see it from my other phone, so I presume that it is visible. So this is the nomenclature of, or this is the structure of the trans and the cis. I told you that the hydrogen that is arranged around the carbon are on a straight chain. So you can see that the cis, the hydrogen, both of them are down, and it is being connected with. There's a double bond that exists between them. Remember, I told you that unsaturated fatty acids always contains an amount of double bond. Because of the double bond, they are not stable. That is why they have been bonded to the chlorine here. All right. Let's look at the trans. You can see that the hydrogens are arranged in an opposite direction. Opposite direction. So, and it also contains double bond. Anytime you see compound that contain double bond, it means that those compounds are not stable. They are not stable. When they are not stable, they try to react with any compound or any molecule that they find itself around them. That is why being able to bond them with these chlorine molecules here. So that is, I just want you to appreciate how the picture look like. So with the cis, they are always the hydrogens that we arrange in a linear form. Once it changes, it means that a lot of properties, chemical properties of it will also change. So some oil, the oil that you can use an oil in a form of cis. When you use it to fry product for a long period of time, the structure will change to cis. And when it becomes it will be in a cis form. When it changes to trans, it has a lot of health implications that we need to be very mindful of. That is why you should be very worried. You should be very mindful how the places that you buy your fried products. Some of those who fry this yam and the rest, one particular oil, they can use it for the whole week. They will use it to fry today, tomorrow they will use it, they will pour it back the next day. The more you heat it to certain temperature, I may not remember the exact temperature, certain temperature, the cis nomenclature of it or the cis arrangement of it will change to trans. And when it becomes trans and it gets to your system, there are three or four diseases that you can suffer from. One of them is blood pressure, because it will deposit inside your arteries and veins. You can also have issue with that to do with stroke. And diabetes people too also have these issues. When you consume a lot of it, it can make you overweight. A lot of fat droplet will be in your system. So that is how I wanted to just appreciate the arrangement. So let me go back to what I was teaching. We are saying that the cis or the trans notation indicate whether the molecule is arranged in a cis. Remember, in a cis, that is a linear. In a cis, is a linear. The hydrogens are arranged in a linear form, in a straight chain. Or in a trans. When it is trans, it means that it is arranged in a branch form. So you can see that the hydrogens are always at the opposite side of them. So that is the difference between the cis and that of the trans. And for instance, if you have six, indicate that there is a double bond on the cis carbon. So when you see this particular notation, this one is telling you there's a double bond, and the 6 here is telling you where the double bond is found. So if it is 6 and it is found, the double bond is found on the cis carbon, you should be able to draw it. So you draw 6 different carbons. 6 different carbons, and there will be a double bond between the 5th carbon and the double carbon. And the arrangement, because it is 6, the arrangement is going to be linear. It is going to be linear. And we said that in case of fat molecules have more than one double bond, the notation may be written as 9. That is, there will be one double bond that is found in carbon 9. There will be another one that is found in carbon 12. So that is what you know. So they may have more than one double bond. So one may be at the carbon 9 and one may be at carbon 12. Please, do you understand the point I'm making? Please, do you understand what I'm explaining now?
So please, can you repeat that for me?
All right. What I'm saying is that there are two main nomenclature when it comes to these unsaturated fatty acids. And this nomenclature is basically based on how the arrangement is done. And I've told you that anytime you see a molecules that contain double bond, it means that they are not stable. They are not stable. Stable molecules always have single bond. Stable molecules, most of them have single bond. They cannot react with any other. That is why unsaturated fatty acids will get spoiled earlier as compared to saturated fatty acids. Sometimes this animal fat, you can keep it for a long period of time, nothing will happen to it. But our coconut oil and this oil that they are liquid at room temperature. When you give it for some time, it will go bad. It will start forming what you call it. Rancidity will start to occur. It will give off flavor, bad flavor. So that is why those things we need to understand them. That with the cyst, the nomenclature, the arrangement is mostly a linear. It's a linear chain with a cyst. Cyst is having a linear chain. But with the trans, they have some amount of branches in it. With the trans, they have some amount of branches in it, and you need to understand that. Then I'm trying to tell you how you can identify where the double bonds are found. Well, we know that those trans and the cis, they all contain double bonds. But we just want you to know that there may be an indication that will tell you that this particular compound contains the double bond and this specific carbon. So we say that when we write a notation like cis, six, well, it is 6 here, and you see this notation as 6. What it is telling you is that that particular compound contains 8 carbon at carbon 6. Do you understand that? It contains 8 carbon at carbon, 8 double bond at carbon 6. And we also say that there may be an instance whereby we can write a notation that there is a cis 9, cis 12. What it is telling you is that that particular molecule is polyunsaturated. It means that it contains more than one double bond. And because it is polyunsaturated, one of the double bond is located at carbon 9, and one of the one of the double bond is also located at carbon 12. So that is what I want you to just appreciate it. The one who has the question, are you okay with that now?
Yes, please. Thank you.
You're welcome. Any other question? Are you all okay? Are you all okay? Hey, Mungasa. Are you all okay?
Hello, sir.
Yes.
It means when they say cis or trans, it means that is double bond, right?
When they say what?
When they say cis or trans, when they say cis or trans, does it mean a double bond?
Cis or trans?
Yes, please. Does it mean a double bond?
No, please. Remember, let me go back. Remember, I told you that unsaturated fatty acids, they contain double bonds. Unsaturated fatty acids contain double bonds. The double bonds can be one or more than one. When it is only one, it is monounsaturated fatty acids. When the double bonds are more than one, it becomes polyunsaturated fatty acids. That is the key thing that I want you guys to remember. So when I got to this point, I said that unsaturated fatty acids can also be put into two main perspectives. We are looking at the arrangement of it, or the nomenclature, or the structure of it. And we know that all the unsaturated fatty acids contain double bonds, all of them, whether being cis or being trans, they all contain. That's that we are trying to understand where they are located, where the double bonds are located. We need to know this because when they are about to undergo oxidation, we will talk about oxidation. When they are about to go oxidation, it is where the double bond is located that undergoes those reactions to cause the fat to get spoiled or to go bad. That is why we need to be mindful, because there are certain things that we can do to prevent this oxidation from occurring. So we are saying that with a cis or trans, we give it a notation. I think this triangle, the meaning of this triangle is, if I remember in our mathematics, it's called delta. So when you hear delta X, it is telling you that the X is just indicating where the number that the carbon double bond is found. And the delta is just showing you the double bond. So that is what it is. So this triangle that you see here, I think in mathematics, the connotation is called delta, if I'm not lying, but I believe it is called delta. So we are saying the cis or the trans notation indicate whether the molecules are arranged in a linear or they are arranged in a branch form. That is what we need to understand. So when we check the cis, realize that it is arranged in a linear form, but when we check the trans, it's arranged in a branch form. That is a few things that I want you to appreciate. Then I spoke about that there may be an instances whereby you can have two double bonds, and immediately there's two double bonds in a particular molecule. It means that that particular molecule is polyunsaturated because the double bonds are more than one. I think I spoke about it earlier before. We say that the double bond is if a double bond, or I said that in case a fat molecule has more than one double bond, then the connotation, this is how we are going to write it. We are going to have cis alpha 9, cis delta 9, comma, and cis delta 12, or cis delta 9, comma 12. What it means is there's one double bond at carbon 9, and there's also another double bond at carbon 12. Do you understand it now? So we are not saying cis contain double bond and trans does not contain double bond. No, when it is only, we are talking it about under unsaturated fatty acids, all of them contain double bond. And you can even see from the picture, this is where the double bonds are. These two lines, these are the double bonds. These are the double bonds. So that is what we need to understand. We're also saying the second point way that we can also name is that cis or trans can also be written in this way. That is W, then you bring your dash and X. A double bond is located on the S carbon, counting from the methyl group. And we say that sometimes the symbol W is substituted with the lowercase n, making it N9 or W9. So it can also be written in this way. When you see the N9 and W9, it is telling you that the double bond is also located at carbon 9, or N6 or W6, or N3 and W3. It is all indicating where the double bond is located. So let me bring this one for you to understand. So these are some of the fatty acids that we've put here. And we are going to look at how the arrangement is done. So you see, we have the butyric acid, which contains 4 carbon. And here, there is 0 here. When there is 0 here, it means that there is no double bond. So the first line is telling you the number of carbon is contained. The second one is telling you the number of double bond is contained. The third one is telling you the position of the double bond. The position of the double bond. So that is what you need to understand, the position of the double bond. So let me come back. Let me look at palmatic acid. Palmatic acid, it contains 16 carbon and there's no double bond. Then when you look at the palmatolic acid, contains 16 carbon and it contains 1 double bond. And the double bond is located on the 7th carbon and it is a cis. The arrangement is cis. It means that it is linear. Let me explain some again so that I will call some of you too. So let's talk about the oleic acid. Remember, the oleic acid is the omega 9. Remember I told you the omega-9. The oleic acid is having 18 carbons. There's one double bond, and the double bond is located on the 9th carbon, and the arrangement is 5th. So I want someone also to pick one of them and explain it, and let me see. Please, I hope you understand how I've given the explanation concerning this particular one. Hello. Hey, move me.
Hi.
Hello, sir. We are here.
We are with you.
Be with me and don't run away. Be like the disciple of Christ. So can you also look at some of them and tell me how we can explain it? Let's look at the.
Please, I want to look at is it linoleic acid? Linoleic acid. It contains 18 carbons, and it also have 2 double bonds, and it's located on the 6th carbon, and the other is on the 9th carbon, and they are all 6.
And when they are all 6, what does it mean?
It means it's linear.
Very good, very good. So in our next one, I think this question we have used it to ask students before. We can just put this there, and we will ask you to explain it. So we can put 18 carbon, then the same arrangement like this, then you ask them to interpret it. So just pay attention to it. That is why I'm spending some time somewhere. Can anyone also give you the type of something different? So there is a arachidonic acid. Can someone talk about it? The arachidonic acid. Can someone talk about it? Hey, where are my people? You people are here.
Sir, what? I can't see from any. Sir, is it the ERUCIC?
Can you see my Keza? Where my Keza is, it's called the arachidonic acid. Can you see my Keza? So just explain that one.
Hey, say that one.
That one is what?
So that one I can't do. Let me explain another one.
No, talk about that one. You can do it. Once you understand, once you know even one, you can explain everything.
Okay, sir. Please, it has 20 carbons. And then 4 double bonds. And then the carbon is on the it is on the 6th carbon.
What is on the 6th carbon?
So the double bond is on the 6th carbon.
On the 6, 9, 12, 15, and they are all
all linear. Yes.
So you see, it's not difficult.
Yes, sir.
Yes, sir.
Who said? Yes, Ula, I want to hear your voice. I want you to talk about one of them. Where is Ula? Or she has run away again. Ula, are you there? Esme, can you speak to one of them for me? Esme, my secretary is sick. She can't talk. Yeah, that's me. I'm waiting for you. Okay, let me continue. Zulea, pick one and talk about.
Let me try one.
Okay, Gloria.
So pronounce the next one.
As you pronounce the next one, which one?
The one after the array like then.
This one is
I can't see you.
Icopentanoic acid. That is the
That's right.
Next one. So
it contains. So that one contains 20 carbon atoms. Then 3 double bonds. 6 double bonds.
Keep quiet. Take your time.
It's 5.
It's not 3.
It's not that one. It's 5. Oh, okay. 5 double bonds. Another 3 double bonds.
Hey, you. You can wait me a week. You can die.
So let me try again.
Double bond.
Let's again.
You can die.
Are you sure?
Sorry, I come again. I don't want you to die along with me, please.
I'll try.
Okay, so sir, it contains 20 carbon atoms. Is that not it?
Yes, it is.
Okay, I'm matching. Then the next one is 4 or 5. I see what.
It's 5.
Okay, so 5 double bonds. Then 3 double bonds, 6 double bonds, 9 double bonds, 12 double bonds, and they are all straight or they are all linear.
Hey, Madam Gloria.
You can die today. Gloria can pass away.
Madam, this one there.
So I can't see the thing where you are trying.
No, the explanation you gave, the problem is not coming from whether you can see or not. The problem is you didn't get the understanding of the
Bond is located on the test is 9, 12, and 15 carbon. Sir, please, can I try it?
Yes, Stella, talk about it.
Okay. Please, the carbon atom is 20, and it's 5 double bond located on the 3, 6, 9, 12, 15, and they are all 6.
Gloria, I hope you heard it.
Yes, sir.
So the problem that Gloria had is after the double bond, once they have 5 double bond, the rest you are just trying to tell us the location of each of the double bonds.
They are located,
the location of each of the double bonds. So close your mouth. Don't talk about that thing again.
Okay.
Amen. Okay. I think Becky, you were saying something.
Amen.
You take the last one. I can hear you. Your voice is far from us.
No, I'm taking the last but one. The docosahexaenic acid.
Okay.
So how is it pronounced?
I can't hear you. Can you repeat yourself? I can't hear you. Oh, no, forget about the pronunciation.
How is the last but one? Yes, please. Hello, sir.
I can't hear you well. Like, I guess.
Hello.
Yeah.
The last but one, the
last but one is
it has 22 carbons and it has 6 double bonds, and it can be found or located on the chassis and
all right, that is good. Nobody spoke about the trans. I think I have one trans in it. That is the ellagic acid, which contains 18 carbon, contain one double bond, and the double bond is located on the 9th carbon, and they are trans. If they are trans, it means that they are primed. All right. So I think when in your quiz or in your exam, when you bring some of this and we tell you to explain them, I think you should be able to do it. But know that, put it in your mind that this trans and the cis double bonds, the body prefers to get the cis instead of the trans, because the trans can cause a lot of conditions. A lot of conditions can be caused by the trans, because the trans can cause a lot of fat deposits in our system. So we need to be very mindful when it comes to that. All right. So there are things here in terms of antioxidants. I can't talk about oxidation without before. I have to talk about oxidation before. What is it?
Please, Madam Becky, can you mute? Your distance is this.
I have to talk about oxidation first before I can talk about antioxidants. So let me quickly pick the slide 8 so that I can talk about oxidation before I can talk about this. I hope you can see my new screen. I've shared a new thing. It cannot be seen. Let me share it again.
Sir, please, it's visible.
No, I'm trying to share something else. Can you see the new screen I've shared, the new one?
Yes.
Can you see that?
Yes. Yes, sir.
All right.
Yes,
No,
sir.
sir.
Lipid also.
So let's quickly talk about lipids of oxidation. Please mute yourself, please. All right. Let's quickly look at the lipid oxidation. Lipid oxidation. First of all, I want you to understand that it is only, or mostly, I don't want to use the word only because I have not read everything. Mostly, unsaturated fatty acids undergoes oxidation. It undergoes oxidation. And remember, I told you that with the unsaturated fatty acids, because they are not stable because of the presence of the double bond. And because they are not stable, they try to react to the environment that they find themselves. So when you have fatty acids, or you have any fats and oil, and you expose it to the atmosphere, it begins to react to the oxygen. The atmospheric oxygen is many, so it will react to the oxygen. And when they start to react to the oxygen, they will produce what you call undesirable characteristics. Undesirable characteristics. So that is what you need to understand. So the more the double bond, the more oxidation that will occur within that particular fatty acid that we are talking about. When the fatty acids contain single bond, they are already stable, so there's no way that you can attach yourself to that particular structure. So we are saying that oil oxidation is an undesirable series of characteristics or undesirable series of chemical reaction involving oxygen that degrade the quality of the oil. So once oxygen attaches itself to the structure of that particular oil, it degrades it begins to cause it to spoil. We say that the important lipids involved in oxidation are unsaturated fatty acids moieties. So remember, I've already said that most of the time it is the unsaturated fatty acids that undergoes oxidation. And because the reason is because they contain double bonds. The saturated one contains single bonds, so they don't have problem. But the unsaturated one contains double bonds. And the example that we have here is the oleic. Remember the oleic, which is the omega-9, the linoleic, which is omega-6, and the linolenic, which is omega-3. These are fatty acids
Sir,
that can.
sir, please, your line went off.
My line went off or yours? Is it mine or yours?
Please, it's yours.
All right. So what I'm saying is that the lipid oxidation mostly occurs, or it is mostly a series. When we say a series, lipid oxidation, we call it a chain reaction. A chain reaction. I'll find a picture of it and show it to you so that you appreciate it. So that the oil, or it is an undesirable series of chemical reaction which involve addition of oxygen to the chain of that particular oil, which degrade the quality of the oil. And when we say the quality of oil has been degraded, our understanding should be that the oil is trying to go through a process that can lead to spoilage, or it may lead to undesirable characteristics such as bad odor, and the texture. The texture may also change. So this is what we need to understand. And we are saying that the most important lipids that involve in oxidation are the unsaturated ones, and I think I have spoke about it. An example is the oleic, which is omega-9, the linoleic, which is omega-6, and the linolenic, which is the omega-3 fatty acids. So these ones, they can easily undergo oxidation because they contain double bonds. Because we just spoke about it, that the oleic contains double bond at the carbon. The oleic, I think it's the carbon, it is 18 carbon or so. It contains its double bond at the 9th carbon. The linoleic also has the same thing, which 8 carbon is also located at the carbon 6. And also the linolenic, which is the omega-3, its double bond is contained or is located at the carbon 3. So the oxidation eventually produces rancidity in oil, which is accompanied off flavor and smell. Or it causes rancidity, which is something that I want you to put much attention to it. And when rancidity occurs in oil, it produces off flavor. So you need to just know that. So that is what we can say about lipid oxidation. But under that, the main thing that I want you to put much emphasis on it is when the specific one that can undergo oxidation. Know that it's unsaturated fatty acid, those that contain double bond. So we say that with the rancidity of oil is the process whereby fat and oil develop unpleasant flavor, taste, texture due to the oxidation. Yes. So when we talk about rancidity in general, we are looking at the process whereby fat and oil degrade. They develop unpleasant flavor. The texture changes, and these are all as a result of possibly the introduction of oxygen into the structure of that particular oil. So we say that accompanied of flavor, that is a bad flavor. Off flavor means an undesirable flavor and smell. So I don't know, those of you that you've done some baking before, have you seen that there are instances that when you put the fat, the one that you use for the baking, the one we call it the magic, the cooked brand and the rest, when you leave it for a while, you will come back and see some mold growing on it. Please, have you noticed that before? Or sometimes it smells bad.
Yes, sir.
You've identified that thing before, right?
Yes,
right.
Please.
So it is as a result of rancidity. It is rancidity that causes that. So we just need to. I just want you to use what is in the house for you to appreciate it. So when it happens like that, it means that there is a lot of oxygen molecules that is within that particular rubber and the oil is reacting to it. It is reacting to it. So we need to just understand that process. So the mechanism, here is another point that you need to just pay much attention to it. The mechanisms that are involved. There are three main stages that are involved in oxidation. Three main stages. We have the initiation stage, we have the propagating stage, and we have the termination stage. So these are things that you should be able to understand them. We have the initiation, the propagating, then we have the termination. The initiation is whereby you are forming the free radicals. There are compounds that we call them free radicals. So oxygen is one of the free radicals that can under the initiation stage. So we have free radicals, we have peroxide, they are also free radicals. Then we have the propagating stage. That is when the free radicals start to form a chain reaction. So what I want you to understand is lipid oxidation is a chain reaction. What it means is that when the product are formed, the product also react with some of the reactants to form another product. Then the same product will come and react with another reactant to form another product. And it continues like that until most of the time all the reactants get finished. Then probably the product will be reacting with each other. When the product react with each other, they start to form desirable characteristics. So you look at them when you get there. So this is a typical diagram of lipid oxidation. So under the initiation stage, there is RH. RH is typically symbol for unsaturated fatty acids. The RH over there is typically unsaturated fatty acids. So these unsaturated fatty acids, because they contain double bonds, and the double bonds allow it to react with the environment. The environment also contains OH. The O over there is oxygen. So the unsaturated fatty acids will react to the oxygen. So at this point, these ones are called the reactants, the oxygen and the RH, which is the unsaturated fatty acid. They are the reactant. Then they will produce what we call the R. The R is a free radical. The free radical, they causes the undesirable characteristics. The free radical. This free radical will continue to react in the environment. So the free radical is reacting with OH. It is reacting with the OH. And it will react with OH. Then the free radical will continue to react with the oxygen again. Then it produces what we call ROO. This ROO is a peroxide. It is the reason why you experience that bad smell and those undesirable characteristics. And this RO will continue to react with the RH again, which is the unsaturated fatty acid, to produce another free radicals. Then the chain will continue. Then the free radical will react with the ROOH, which we call the hydroperoxide. So you can see that when the reactions occur, the products come back to react with some of the reactants. And sometimes even the product react with the initial product that has been produced before. Then those processes will continue, and it will continue until the bad smell is felt, or all the bad characteristics is felt. When it does that, I will come back to the initial stage and everything. I will explain them again. When it does that, it moves straight to it will get to a point that all the fatty acids will get finished. It will get to a point that you can't even get some of the fatty acids again, or the unsaturated fatty acids again. So what will happen is, because there's no more unsaturated fatty acids again, what will happen is the free radical that has been produced will begin to react with each other. That is where the termination comes in. It's like you have a negative something that is reacting with a positive something. At the end of the day, it will come and produce a positive result. So when you have negative and negative, it will give you positive. Is that also?
It's so, sir.
So let's say you have a free radical, two free radicals that is reacting with each other. It is going to produce what we call RR, which is a desirable characteristic. That is where the termination stage comes in. So let me pick it one after the other. At the initial stage, that is where free radicals form at the initial stage. Put it in mind. And the free radicals are formed as a result of the reaction between the unsaturated fatty acids and the atmospheric oxygen, and to produce free radicals. That is what you need to know. So when the free radicals are formed at the initial stage, we quickly move on to, and the free radicals, we quickly move on to the propagation stage, whereby these free radicals are going to react with the unsaturated fatty acids again to produce more free radicals. So a lot of free radicals will be produced, and each of the products that has been produced will also come back and react with the reactant and that of the free radical that has been produced previously, and to continue like that until the oil gets spoiled. It will get to a point that the oil will get spoiled, or the fat will get spoiled, to the point that there will not be more unsaturated fatty acids again. And when there's no unsaturated fatty acids again, because the free radicals are many, it will move to the termination state. Termination means that you are trying to stop the reaction, you are terminating it. So under the termination state, most of the free radicals will start to react with each other, and they will begin to produce desirable characteristics. So that is what I want you to just understand. So let me see what the notes try to say. So we are saying that the RH is unsaturated fatty acid. R is a free radical formed by removing a liable hydrogen from a carbon atom adjacent to the double bond. And you know, anytime a double bond is formed, one particular hydrogen is lost. I don't know. Let me see if I can get this, because if I don't show you a picture of it, understanding it become quite difficult. I'm coming. Let's give me some few minutes. Let me see if I can get it, so that you can appreciate it better. Let me share this particular thing for you here. This is a slide I use for my sandwich students. So let me see. I hope you can see the new slide that I have shared. Can you see it? Can you see Unit 4, lepers? Can you see?
Yes, sir.
Yes, sir.
Correct. Easy. So let's look at it. This is the picture that I'm trying to. I want you to look at it. So you see this one. This one is lipid oxidation in an oleic acid. Or this one, no, this one is a monoic acid. So we have there's a lot of carbons. They didn't draw all of them. You just give us the 7, the 8th up to the 12th one. There may be others that are there. It contains one double bond, and the double bond is found in carbon 9. So carbon 9. So this one is an example of omega-9 fatty acid that contains the 18 chain, if you remember. So you see that at the normal circumstances, at the carbon 9, there was supposed to be a hydrogen over here. And the carbon 10, there was supposed to be a hydrogen over here. But because of the double bond that is found, those hydrogen has been lost. And when these hydrogens are lost, because if the hydrogens are there, it gives it a rigidity, it will give it a stable nature. But when the hydrogens are lost, this particular compound is not stable. So they try to find stability. And how do they find stability? They try to react with other molecules or other compounds around where they are. And because you have exposed it to the atmospheric oxygen, oxygen will come and attach itself to the carbon 9. When oxygen attaches itself here, it starts to develop these undesirable characteristics that I have made mention of, the bad odor and the rest. That is how it is. That is the point I was trying to explain. So that is why we say that the free radical formed by removing a liable hydrogen, that is, the hydrogen has been removed from the carbon atom adjacent to the double bond. We are saying that ROOH is a peroxide, one of the major initial oxidation products that decompose to form compound responsible for the off flavor. So I told you that these free radicals are those that cause the off flavor. And a typical example is the hydroperoxide. So when this hydroperoxide is formed, it decomposes because they are volatile compounds. They can easily decompose that they are broken down. And when they are broken down, they give that particular odor, the bad odor that you see. And we said that such secondary products include hexanol, pentanol, and melanolaldehyde. So these are some of the secondary products that can be produced when these reactions are being occurred. So this is a typical example. This one I have explained it. We said that hydrogen could be removed from carbon 8 or carbon 10. That is adjacent to the double bond. So you see that the carbon 10 is the one that has been removed, rather than carbon 8 one is there. So let's use another example. So you see this particular one, because it is going through oxidation, the hydrogen that was removed, it has indicated. So this point where it has indicated the hash, this is where the oxygen is going to attach itself. And when oxygen attaches itself here, then it starts to develop all these free radical things that I've been talking about. So you see, here, the double bond has been shifted from carbon 9 to that of carbon 10. And when the double bond shifted from carbon 9 to carbon 10, look at what has happened. The hydrogen has rather been lost by carbon 9. So that is how the reactions occur. And they are series. I record this one as chain reaction. A chain reaction. It means that the product will continue to react with another free radical in order to produce a lot of undesirable characteristics until it gets to the termination stage. So there are a lot. I think when I have to send you this particular slide so that when you are reading, you appreciate the structure. I'm just using the structure for you to get a lot of understanding. So you see here, this particular one, there's OO here. So you can see that the addition of oxygen. So 2 oxygen has been attached. And that is, it has formed a peroxide. And it's a radical. It's a free radical. And this free radical will cause a bad odor or undesirable characteristics to the product that it found itself. So that is the picture that I wanted to.
Sir, please, you can't see it well. It's too small.
It is, you can't see it well. Like how?
Yes,
Is
on
it
the
too
screen.
small or what?
When the thing has shifted to, it was supposed to be on the screen, but
Is it not on the screen?
no, it looked like somebody's page, something like that. Too small. Yes.
Is it the same to everybody, or you? The rest, is it the same, or because I can see it well here.
I can see.
It's the same. Move to the top corner. Just at the corner.
That's moved to the top corner. Wow. Are you using phone or laptop?
Phone.
Maybe it's a phone. We are also using phone, but we can.
I'm using one tablet. I'm rather sharing it on my laptop, but I have a tablet that I use to monitor whether it can be seen or it cannot be seen. So probably you can adjust your phone, or you can do something about it to see it well. Because I don't think there's a button here that I can use it to adjust. All right. Please, do you have any problem with whatever that I've explained? I know this one, they are not quite familiar with you. but I think I was trying to just break it down for you to just appreciate it better. Are you okay with it? Hello? Hey, move here. Are you okay with it? When it comes to liquid oxidation, especially the stages, the 3 stages, are you okay with that?
Yes, sir.
So put it in your mind that you have 3 main stages. The initiation stage, that is where you form your free radicals. The propagating stage, that is where your free radicals will start to react. Then we have the termination stage. Under the termination stage, that is where this free radical will be reacting with each other to produce a desirable effect. So you need to know that. And we are saying that there are certain things that can speed up the reaction, and one of them is copper. There are certain metals that can speed up the reaction. We say that copper did not initiate the reaction, but once hydroperoxide were formed, it speed up the breakdown. So you know the hydroperoxide are the one that is responsible for giving out the bad odor. So copper will help you to break it down. So that, and when copper helps you to break down, it means that it is facilitating the process. It is facilitating the process. So that is what I want you to understand. We are almost done. Oh, we are almost done. So let's see how. And there's one thing I want to let me
Same. Same. Sir, please, the stages you mentioned, I didn't hear. I didn't hear it all. I heard you say that we have 3 stages, but I don't know 3 stages. Or you mentioned the oxidation stage. I didn't hear it, please. I don't know whether you can help me.
I said the stages, we have the initial stage or in the initiation stage. We have the initiation stage. We have the propagating stage. Propagating stage. Then we have the termination stage. The termination stage. So these are the stages that I made mention of. Are you okay? And I told you that with the initial stage, that is where there will be a formation of free radicals. There's a formation of free radicals, and under the the propagation state, that is where the free radicals will be reacting to form the peroxide and the hydroperoxide. And I told you these are volatile compounds that when they are formed, they can easily be decomposed. And when they are decomposed, that is where we are able to see what we call we are able to have a feel of the smell, the bad odor, and the rest. So that is what we need to just understand. So we say that we have the initiation stage. That is it involves the formation of the free radicals. So that is one thing that you need to remember, that at the initiation stage there is a formation of free radicals. And with the propagation stage, that is one free radical. Once they are formed, they react with other molecules. Particularly, one of the molecules that the free radical reacts with is the unsaturated fatty acids itself. When they react with the unsaturated fatty acids, then they create a new free radical. And I told you that this process is called a chain reaction. They produce a new free radical, and the process is called a chain reaction because the product will be reacting with other radicals to form a more complex compound. Then we said I have the termination stage. I said that at the termination stage, when the propagation phase continues until all the free radicals are deactivated, then effective it got to a point that it will stop because the free radicals will be reacting with each other. So when the free radicals act with each other, all the free radicals will get finished. And when the free radical gets finished, that particular action will be terminated. It will be terminated. So that is what we can say. We can also use antioxidant to neutralize these free radicals. We will use some antioxidant to neutralize this free radical, and it can even start from either in the initiation stage or probably when you get to the propagation stage, you can use them. So that is why I want us to look at free antioxidant. But there was this question I wanted to ask, that when you have monounsaturated fatty acids and polyunsaturated fatty acids, which of them will have more effect or severe effect as a result of oxidation? Which of them that the oxidation will be many or it will be very common? Which of them? Do you understand the question? I'm saying that you have monounsaturated and polyunsaturated fatty acids. That is, the mono one contains only one double bond, and the poly one contains more than one double bond. Which of them will undergo oxidation faster? And why? When you tell me which of them will undergo oxidation, then you tell me why. The one who will get this question, I think I'll be very happy. It means that they are making progress. Yes, which of them will undergo oxidation faster and why? Hey, I have 43 people here. Let's talk. Which of them? Or if you are not able to differentiate which of them will undergo more, it means that you didn't get some of the concepts right. Which of them will undergo oxidation faster? Yes. Let me open the names and mention people's names. You cannot just be here and keep quiet like that. Yes. Who is talking? Who is talking? Hey, no one. And I'm on to you don't hear me.
We can
Or you didn't hear my question. I'm going from the silence. It means I'm teaching in the cemetery.
No, we are
Monosaturated and polysaturated. Yeah. Yeah, Zulea, give it a try. Hey, I never knew you people can just turn the online class into a cemetery like that. Only one question. So what will make let me put it in different way. What will make a particular fatty acid to undergo oxidation? Yeah, what will be the key thing that will make a particular fatty acid to undergo oxidation? Yeah. Janet, you wanted to talk?
Is it the bonds in between them? Hello,
Rebecca,
sir.
come again. Yes.
You are asking what key characteristics will make unsaturated fat to go under oxidation? And I'm saying that is it the bonds, because one has one bond and then the monosaturate has one bond and the poly has two bonds.
And I'm talking about unsaturated. So here, when we are talking about lipid oxidation, we are not even looking at saturated fatty acids. They don't have problem with issue of oxidation. It is the unsaturated ones. And I told you people that they go through oxidation because of the presence of double bond. And when a double bond is found inside their structure, it makes it loses hydrogen. And when hydrogen is lost, it is no more stable. Once hydrogen is lost, they are not stable. So they try to find stability by trying to react with the molecules or compound that they found within the environment that it found itself. And I'm asking that with the monounsaturated fatty acids, they contain only one double bond.
Mm-hmm.
And the just listen to the question well, you'll be able to answer. And the polyunsaturated fatty acids contain more double bond. And I'm asking that which of them will undergo oxidation faster? That is my question.
So please,
the poly one.
The poly unsaturated has.
The poly one.
Yes.
How many people support her with the poly one? I want to know those who support with the poly one.
So the mono one, the mono unsaturated.
Remember I told you, I said that which one will undergo
so the poly one
why?
because it contains oxidation faster.
It contains oxidation faster.
It's because of the double bonds in the poly one.
So
It
please,
contains double
mono,
bonds which are more vulnerable to attack by oxygen.
so please, to my understanding, the mono one would be.
Patricia, tell me the reason why the mono one will undergo oxidation
Please,
faster.
to my understanding, the mono, it has only one bond. So I think that will be faster compared to the poly, which contains more double bonds and other things.
So those who are also saying it is the poly one, I want to also counter her with your explanation. Yes. Who believe it's the poly one? So that you can also counter her with your explanation. Then when you people are done, I will tell you the specific one. Yeah, Gloria.
So the poly one, because of the double bonding, is being exposed to the free radical reactions.
Okay. Any other explanation to that? I think Becky, you wanted to say something.
No, I was saying that it's the double bonds because they have the double bond, they are always vulnerable.
Becky, take your time. All of them contain double bond. Just that one contain one double bond and other contain more than one. So choose the words carefully. Yes. If not, when you're explaining it in your book, you may make a mistake. You may know the thing, but how you put it may go against you. So Becky, come back again.
So with the poly one, it has more double bonds. So in this case, it becomes so vulnerable to be attacked by the oxygen than the mono. The mono one, it has just one double bond. So the attack will not be so much than the poly. The attack will come, but it will not be too quick like than the poly. So that's how I understand it.
Oh, okay. Well, Vida, you wanted to say something. Vida.
Hello, sir.
Yeah, Vida.
Please, I wanted to say that with the poly, it has more double bonds, and that it has lost a lot of hydrogen atoms. So it makes it reactable. I don't know if the word is right. It can react with so many. It's not stable, so it can react with so many things at a time. That is why I would choose the poly to the mono.
Okay. Any other explanation from any other person? Martha, I saw your hand.
Yes, sir. Sir, I will say the poly because it has a high number of double bonds.
And when you have high number of double bond, what makes it go through?
Sir, let me take it from there. Since it has high number of double bonds, each of these bonds will react with the free radicals, and as they react, each of them it will continue to react and matter.
Oh, maybe I don't know what to
say.
I'm enjoying the explanation. That is why I keep on asking different one. Who said?
The more the bonds, the more the reactions takes place. So it's the poly one.
Okay. Thank you.
I think Patricia. Is Patricia around? Patricia.
Yes, so I'm here.
Nobody supported you. You are Uncle.
Yes, so.
So, Patricia, I think your colleagues have been able to convince you, or they have confused you. Which one?
It's like they have convinced me.
Very good. Once you are convinced, and I'm happy that most of you are, and I guess why I'm happy that you've been able to answer this question. Once this question has been answered, it means that that point is well made, and it is obvious. The only thing that I will say is what your colleagues have said, that the more you have the double bond, the more you are exposed to these oxygens. And when you have only one double bond, probably it's only one oxygen that can be attached to where the double bond is. But here is the case, you have about 4 or 5 double bonds, so a lot of oxygen will be attached. And when more oxygens are reacted to your system, you are going to produce more free radicals. And when the free radicals are many, it means that the undesirable effect is going to be many. It's like you have a lot of sore on your hand, and there are about only one sore at your right hand, and your left hand there's about 10 different sore. You know, a lot of bacteria can enter the one that have a lot of sore compared to the one that have only one sore. So that is some of the scenario we can create. So the polyunsaturated fatty acids undergoes oxidation faster as compared to the monounsaturated because of the number of double bonds that are involved. Amen. I'm happy.
Amen.
I'm happy.
Thank you, sir.
I'm happy. Like I'm seeing A1 and B2s and the B3s in your responses. That one there, you have to learn, because God doesn't set questions in chemistry. God will only set questions in Judgment Day. So now let's look at
Hello,
oxidation.
Sam.
Yes.
Please, you said we shouldn't talk about network, but I'm sorry.
Hey,
Please send a call.
Ramatu, where are you?
So I'm in Krachi, Oti region.
Your place is too far. Can't you travel to Accra?
Hey, sir. Please, the question, I don't know whether I got it right. Like, what key characteristics will make unsaturated fatty acid to go under oxidation? Please, is that the question?
It is similar. I was asking, yes, the first question was, what were the key characteristics? Yes.
Hello, Sam.
The answer was
please,
because of the presence of double bonds.
Please, is that the question?
Yes, the first question. I asked 2 questions. The first question is the one that you said.
Okay.
What key characteristics? And the answer was the double bond.
Okay.
And I came back to ask that the unsaturated fatty acids, you have 2 main types, the mono one and the poly one. And I asked which of them will undergo oxidation faster.
Okay.
And the whole class said it is the polyunsaturated because the double bonds are many. And the more the double bonds, the more it is going to react to the oxygen that is found around its environment.
Thank you, sir.
Thank you, Madam Kratchik. All right. So let me quickly talk about, I think I have some few minutes. Let me quickly talk about how are we going to prevent this oxidation from occurring? How are we going to prevent this oxidation from occurring? You know, we have antioxidants. Anytime we have, in computer, we have viruses and we have antivirus. It means that when there is a virus, the antivirus can deal with it. So once we have oxidation, there is also an antioxidant that we can use to deal with this oxidation, because most of the oxidation that occurs in our lipids give out a bad effect. So we want to do away. So we say that there are substances that inhibit oxidation, especially one used to counteract the deterioration of stored food products. Antioxidant function by interfering with the chain reaction. If the number of free radicals can be kept low enough, oxidation will not occur. So you see a point that we are making here. We are saying that these antioxidants, their work is to inhibit oxidation. And this oxidation and they do it by trying to counterattack any form of deterioration that can be that can cause stored food. And we are saying that these antioxidants, they function by interfering the chain reaction. And you know, we've spoke about the chain reaction, that oxidation is the chain reaction. That is from the initiation, from the propagation. The last one will be the termination. So how do they do that? They try to keep when we are saying that when you keep the number of free radicals low, oxidation will not occur. Let's say you have a whole of fatty acids and probably one free radical, only one. Probably when they are low, their reaction will not be seen. Their reaction may be going on, but it might not be seen because they are low. But when they are more, it's like before you fall sick, or before the doctor will say that you are suffering from malaria, it means that the plasmodium, there are many in your system. When they are few, you may not feel the effect of it. But when they become many, that is where you feel like you can't eat. Your body, you feel tiredness, the headache, you'll be sweating anyhow. So that is how the scenario that we can also use to understand this concept. And say, in order to function well as an antioxidant, a molecule must, one, react more with the free radical rapidly than the free radical react with the oxygen. So you say this point, when you are adding any substance to a fat or a lipase to stop the reaction or to stop oxidation, that particular substance should react more rapidly with the free radical than how the free radical reacts with the lipase. Do we understand this point? Do we understand this point?
Yes, sir.
Hello?
Yes, sir. Yes, sir.
So we are saying that if the antioxidant you are adding to the product can react, maybe in 2 minutes it can react with the free radicals, about 10 free radicals, and in the same 2 minutes the free radical can react with only 4 fatty acids. Then the free radical, then it means that within a short time, this particular antioxidant can react with most of the free radical, and it will not give it, or those radicals will not be available for it to react with the lipid that will cause it to spoil. So that is the point. We are saying that. So these 3 points is very, very, very important. Pay attention to it. We are saying that the product of the reaction with the free radical must not be pre-oxidant. That is another point that you should also understand. That the product should not be pre-oxidant. It means that when the free radical, when the antioxidant reacts with the free radical, the product that is going to form should not also be a product that can undergo oxidation. That is what it means by pre-oxidant. Cannot undergo oxidation, because if it undergoes oxidation, then there's no need, because it will continue to work. And the last one is the molecules might be lipid soluble, because if it is not lipid soluble, it cannot be. So it means that the molecule should be hydrophobic in nature, because lipids, if you are not hydrophobic, you cannot work within lipids. Please, do you understand these 3 points? So these are the 3. I can ask you that state 3 characteristics of a good antioxidant. It's a question that can be asked. Characteristics of 3 antioxidants. The first one is it's supposed to be react rapidly with the free radical than the free radical reacting with the lipids, and also the products that is formed from this reaction must not be pro-oxidant. And the last one is the molecule must be lipid soluble. Are you okay with that? Are you okay with that?
Yes, sir.
Can I continue?
Yes.
So we said that there are 2 main types of these antioxidants. I think I'm done. So we have the synthetic one and that of the natural one. So we have the BH, the synthetic one. We have the BHA, that is a butylated hydrogenol. So we said that this may hinder the effectiveness in vegetable oil, but increase the carry-through potency for which BH is known. So we can use this one in vegetable oil, and the use is We have the lard, the shortening, the vegetable oil. You know, lard is the fat that we get from which animal? Lard. From which animal?
Cow. Cow.
No, not cow.
It's from pig.
Pork. Pork is the name of the meat, but the pig is the animal.
From pig.
So am I right? Pig.
From pig. If you say from pork, yeah, I won't give you your max. I'll give you half. Then you also have PG. That is the prepared garlic. Is also another form of synthetic one. So these ones you can read them. Let me see if I can see the natural one. So the synthetic one, there are many. There are many, and there's they are also saying that there's a situation whereby you can combine some of them, and one of the reasons is when you combine them, you take advantage of the different properties because some of them may work better than the other. So and also allow better control of accuracy. They may provide the synergistic effect, and also combination may provide more complete distribution in food more than the other. So what they at this point is trying to tell us is when you are trying to use antioxidant, don't just be extremely glued to only one. Probably you can use different types so that the potency that this one is lacking, the other one can produce. All right. So this ones, I think my time is up. These ones are all reading things that you can read. So the natural ones, you can get some from oats. Oats contain a lot of over 25 phenolic compounds. The phenolic compounds are some of the properties that they are found in it. And we can also derive some from caffeine and ferulic acid. So alternative to antioxidants. If you don't want to use antioxidants, this is one way that you can do it. Elimination of oxygen. That means that you are going to package the food in an environment whereby oxygen cannot get access to it. So sometimes you use nitrogen, or you do a vacuum packaging, or you can do packaging and add what you call oxygen scavengers, those that can find oxygen. So this is what you can also do. Then also you can also eliminate elimination of sensitive substrate. That is replacement of polyunsaturated oil with less saturated oil. So you see this point. When you are producing a product and you think you want to keep it to avoid oxidation, try and use a lot of less unsaturated one. So probably the one that contains mono unsaturated one should be used more than the poly one, because we all understand that the poly one undergoes oxidation more faster as compared to the mono one. So let's always go in for the mono unsaturated ones if only that is what we want to use. And also decrease the rate of oxidation, that is storage at low temperature, because most of the time oxidation like a higher temperature to do. So you slow down the temperature and also you store it in the dark places. If you have time, read some of the things that facilitate oxidation. One of the things you get to know is light and some metals. So when you store them in a dark area whereby light cannot get access to it, you may be able to do away with oxidation. Then we say that use fat and oil that contain low level of oxidation promoters. Example is heavy metal. I think I have made mention of that. A use of ingredients that are naturally rich in antioxidants. Some ingredients automatically contain antioxidants. So the oat that I make mention of is one of them. And also we can also do treatment that we call hydrogenation. That is treatment of oil with hydrogen and a suitable catalyst to decrease the number of double bonds. So the more you are adding hydrogen to it, the more you are reducing the number of double bonds. So that is what I can say. And we have close. So the effect of hydrogenation, when you add a lot of hydrogen to unsaturated fatty acids, they become saturated fatty acids. When you add a lot of hydrogen to the liquids, it becomes solid. And when it is cis, you add a lot of hydrogen, they can become cis, or the other way around, they can become trans. Mapon, any question on your microphone? Let's have our last interaction, then we are done for the semester. Mapon, I have closed.
Sir, please, sir. The three characteristics of
oxidant. So I didn't get that place well. I beg you, sir, repeat it for me.
Oh, Martha, my bro. We are saying that. What we are saying that is for antioxidant to work well, or a pure characteristic for antioxidant to work well. One, the antioxidant should rather react faster. It should react faster with the radicals, the free radical, than how the free radical reacts with the lipids. Do you understand that particular point?
Please, yes.
That is one. Because if the free radical rather reacts faster with the lipids than the antioxidant, then the antioxidant cannot work. Its work will be slow, but rather it's supposed to be faster. That is one. Two, before an antioxidant can work, it should be lipid soluble. If you cannot dissolve in lipids, how will you even be in the lipids and react and stop the the free radical? So you must be lipid stable. That means soluble. You should be able to dissolve in lipids. That is second one. And the third one is the product that will be formed after the antioxidant has reacted with the free radical should not be pro-oxidant. It shouldn't be a product that can undergo oxidation. Pro-oxidant is some substances that can undergo oxidation. We say pro-oxidant. You know, we have certain vitamins that we call them provitamins. It means that they are not vitamins on their own, but when they get to our body, the body can use it to provide vitamins or synthesize vitamins. There are certain products that they are not oxidant on their own, but they can undergo oxidation. We call it pro-oxidant. So these are the three characteristics. The results or the product that will be formed between the antioxidant and the free radical should not be pro-oxidant. Then the antioxidant should also be able to dissolve in lipids. That is the second one. And the first one that we spoke about is the rate or the chemical reaction, the rate of the reaction between the antioxidant and that of the free radical should be faster than what the free radical does with the lipids. Are you fine now?
Yes, sir. Thank you very much.
You're welcome. I saw another hand up. Any question? You can on your microphone now. Any question? Read the notes. If you have any challenge, give me a WhatsApp chat probably. Yeah, Cynthia. Cynthia.
Hello, sir. Please, mine is not a question.
Supply something with a liquid. Sir, I'm talking. Oh. Hello, sir.
Yeah, the one asking the question, I don't think that's well. Yeah, come again. Yes, come again. I want to hear you well. Yes, Cynthia, are you the one talking? I can't hear you.
Sir, please, can you hear
Yes,
me?
I can hear you now. I can hear you.
Okay.
Oh, Cynthia, I don't know what is happening. Cynthia, I can't hear you again. I can't hear you. Maybe you have to reposition yourself and come again. Please, any other question? The whole class.
Sir, please, my question is it good for you to use an oil and leave it so that the next day or the next time you use it again? Is it?
I don't know. I spoke about trans fat. I spoke about trans fat, and one of the ways that trans fat is developed is the overuse of a particular oil. That is, when you fry a product today, then you leave the oil, then tomorrow you use it. So I will set for the actual temperature that when the oil is heated up to that point, it becomes trans. I'll find out. But you can also read it. But what you are saying is it is not good. It is not an ideal situation that one oil will be used over and over and over again. But that is what those people who have been frying the kose, the yam, and all the fried products out there, that is what they do. That is what they do. So most of those food that we eat contain trans fat, and this trans fat, when they get to our system, it causes a particular problem that we call atherosclerosis. That is when there is a deposit of fat deposit inside your veins and your arteries, and that is what causes a lot of blood pressure, because it blocks your arteries. That the number of or the amount of oil that passes through your arteries will be decreased. So your heart need to pump it harder before it can pass through. And when it continues for a long period of time, it can cause a stroke. That particular disease can lead to stroke. So people who are having pressure, before you get stroke, a lot of them, they get pressure before they get the stroke. And those people who are fat, you are also at a disadvantaged position, because once you are fat, you are already prone to getting those kind of problems already. So we should be mindful of how we may not be able to control those who sell outside, but probably in our houses, oil is something that we should take it minimal. We should reduce it. I don't know whether I've informed you. When you pick one gram of oil or 1 gram of fat, the energy that it gives you is 9 kilocalories. I think we will do this probably next semester, if I will be the one that will be teaching you that course. You will do applied nutrition. I don't know the center that I will be teaching. You did it in science. Oh, okay. Okay, so one gram of...
We did it in nutrition.
Okay, yes. 1 gram of fat will give you 9 kilocalories. But 1 gram of carbohydrate and protein will give you 4 each. So you can see that oil is very rich. Oil is very rich in energy. So it can, when it is stored in your system, you should be worried. It is not something that should be stored in an excess. Although the body needs oil to protect certain organs, like your heart. Under our skin, there are oil that regulate our body temperature and the rest. But when they are in excess, it becomes a problem. So trans fat is not the best. So we shouldn't be reheating oil over and over and over again. But try and find out the temperature level. I think it's something that it will benefit all of us. Yeah. Any other question? Any other?
Please, I wanted to ask if you will meet next week.
No,
for Samuel.
Yeah,
Please. Let me so that you summarize everything for us. Especially the unit 1. The DNA.
The summary is inside the slides.
It's very important.
We beg you.
The grass and the one. I bought bundle this morning. It had finished. So the timetable said I should teach you up to today, and I have done that. And I think it has gone well, or it didn't go well.
You just want something. We'll buy you the distance, the data. My people, are you there?
Yes, there we will buy. Yes, we buy.
Please, they are recording the class, so please.
Don't mind them. When they call them, they'll go and answer their questions. All right. So basically, I think we are done. We are done. And unless probably you have a special situation that you need help. Aside that, I think I have taken my time to the best of my knowledge. I think I've handled the class in a way that I want everybody to just appreciate whatever that we did. But if in the course of the week, if people have any challenge, come together. I think we have a platform, so probably we can communicate over there and find a way forward. So those of you that you are not on the platform, probably you can contact me personally, then we know what we can do. But if I will organize another class for you to get a better explanation, let me see a week. I'm also very tight person, The whole of the week I will be on UCC campus doing 1 or 2 things to do. We will see what you can do. But if you read the notes, if you have any challenge, if you come across any question that you are finding difficult, put it on the platform when I see it. And I'm very reactive when it comes to phone. And my phone is always with me, so I will quickly respond to it. So you don't have problem in that.
Okay, sir. Thank you.
Vida, you can write this number down and you can WhatsApp me after the class. I will forward the group link to you. Vida, you are asking that you are not on the platform. The number is where is Esme? Esme, what's your number? Esme, can you talk? Esme.
Then let me see if I can get it for her.
Get Esme number for her so that she can. If you can't find Esme number too, you can also the class is also saying we should add it to the platform. Yeah, I'm not that it is not feeling well. So those of you who are on the platform, add those people who want to be on the platform number to the platform so that we can just add them to it. Maybe when your number
is 0546
0546
83
51
36.
36.
Yeah.
Vida, are you okay? Vida and Ketekrachi. Oti,
Yes,
are you okay? Oti, Ramatu Oti.
Yes, please.
Oh, okay. I think you have microbiology after this class, right? So let me allow you to just have some rest and prepare for the next class. No matter what will happen, if I'll meet you guys again, I will let you know on the platform. So without any question, then I think have a good day. Bye bye. Hey, Mongaza.
Bye.
bye bye.
Bye. So thank you very much.
Bye bye.
Have a nice weekend.
Thank you so much, sir.
A blessed one.
Welcome.
Greet your wife for us.
Oh my.
He doesn't have. He doesn't have. So if you can give me one.
I want to. Gloria, you are selling me on this platform.
Okay, sir.
Let me go.
Ula, where is Ula?
Oh,
I didn't invite you.
I didn't want to come in.
Hello. Hello, colleagues. Hello.
Yes.
Yeah, please, which teacher takes the microbiology? Is this Israel? Hello.
Depends on your center.
I'm talking about the general one, those who do the online solely. Today, my leg is paining, so I couldn't go.
So you can join Sir Israel.
Oh, okay. All right, thank you.
Yeah. Okay.